So, hello students, welcome to Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. Good morning, each and every one of you. Right, students, as the examination is coming near every single day, we have decided that whatever backlog you will have in your entire preparation of your NEET examination, it is going to be clear in the next few days, right? So, for the very same reason, I believe this is a chapter which is a backlog to many students because there are so many concepts here in this chapter. It becomes a little difficult to visualize it. Yes or no? Right? For example, if I tell you diathecus, bilobed structure, if you read the lines of NCERT, it can get a little confusing. But if you visualize this, it becomes even more amazing. So that is what we'll be doing in today's class. Any student who has zero knowledge, absolutely zero knowledge in this chapter, you will learn the entire chapter at the end of it. It could be the concepts like, sir, what is porogamy, what is chalazogamy, what is mesogamy, what is monocolpate, what is bicolpate, right? There are so many concepts to learn here in this chapter which is generally overlooked, and I'm going to make sure you learn the entire out of it. Okay, so students, good morning. Yes, we'll be solving a lot of questions. Do not worry. Yes, there will be PYQs. I have PYQs today. Yes, I have PYQs, so you don't have to worry about it. This is going to be a breeze walk. Now, let me ask all of you a very simple question for all of you. Right? What comes to your mind when you think of a flower? In the chat, all of you. Good morning, everyone. Good morning. Tell me, students, what comes to your mind when you think about a flower? Like, generally, students, if I say flower, what exactly is coming to your mind right now? For example, recently Valentine's Day happened. If you love someone, generally the feelings are expressed with the help of a flower, a rose or whatever favorite flower is. If you're a very religious person, if you pray every single day, we know that we put flowers, we give flowers to the God. So flower has a lot of different values to express your feelings, to the religious values, or for ornamentation. For example, imagine a house like this with beautiful variety of flowers. Isn't that amazing? Yes, it is. Now, can anyone tell me in the chat, students, what is the main reason for this variety of flowers? Like, how do we have such variety of flowers? Because if you want to have variety, do you need asexual reproduction or do you need sexual reproduction? That is my first question to all of you. To achieve this variety of different types of trees, different types of flowering plants, the angiosperms, the main reason for such variety is sexual reproduction, students, sexual reproduction. Now a question to all of you goes to all of you right now. Right? I told flower this, flower that. I told you flower is this, flower is that. But for a biologist or a botanist, can anyone tell me what is a flower for a botanist? For a botanist like me, right, flower is nothing but a reproductive organ. Flower is nothing but a reproductive organ for me. Apart from that, students, if I have to tell what is a flower, I will call it as a condensed shoot. Now, students, you might have heard this line, sir. Flower is a condensed shoot. That is, I can also call it as a modified shoot. Yes or no? But what is the meaning of this? What is the meaning of a modified shoot or a condensed shoot? Let me tell you with the help of a diagram. I want everyone to focus in my class right now because it is going to be very technical. Any student who has zero knowledge will gain the utmost importance in this chapter. Listen to me. Now, if you know basic botany, right, you can tell me this is a stem. Let's say this is a stem. On this stem, I have this small lines. What do I call these lines, students? What do I call these lines? Can anyone tell me? What do I call these lines on a stem? Let's say I will call them nodal regions. Node. Okay, this is a node. Between two nodes, this gap right here, this one is called as internode. It is called as internode. Okay. Now, at this internode, what happens, students? At this internode, generally what happens is, there is a leaf. Yes or no? At the internodal regions, at the nodal regions, we can generally find a leaf. We can find a leaf at the nodal region. Now students, if you all of you remember from the chapter anatomy or morphology, I always told you at this nodal regions we generally find a bud here. Can anyone tell me what is the bud here? The bud which we find here is the axillary bud. Yes or no? Axillary bud. Now this axillary bud in future it can arise into a branch or a flower. Let's say it arise into a branch. It is arising into a branch. I'm gonna draw it one more here. Let's say it's arising into a branch like this. Arising into a branch. Now what happens here is I want everyone to focus really well. All of you see this branch? Yes sir, we can see the branch. We can see the branch here. Now students, what if this long branch, right? This long branch which has nodes and internodes. What if I condense the branch? What is the meaning of condensing here? The meaning of condensing here, all the internodes becomes very tiny. All the internodes becomes very, very tiny now. All the internodes are reduced. Let's reduce them. Let's reduce them. And all the nodes are together here. So we end up getting a structure, something like this. Something like this. Now this condensed structure right here, condensed structure undergoes certain modification. This condensed structure now undergoes certain type of modification. Into a structure like this. Can anyone tell me what is the structure now? This swollen end. This swollen end, can anyone tell me what is the swollen end? I'll tell you, don't worry. I'll tell you, don't worry. Can anyone tell me what exactly is swollen end here? Anyone? Because this swollen end right now, on this swollen end, will generally find our flower. Anyone? I believe all of you are understanding today how the flower actually develops. We have something called as flower primordia. This flower primordia will generally become, shoot gets completely condensed. The shoot gets completely condensed here. And can you see, this is a stem. It has something called as a leaf, axillary bud. The axillary bud develops into a branch or a flower generally. This is a branch here. If this entire branch is getting condensed, condensed shoot. It is a shoot. The shoot is getting condensed. The condensed shoot is undergoing modification. Undergoing modification. And this structure right here is nothing but the thalamus. The swollen end of a flower is called a thalamus, and on top of thalamus we can see the gynoecium, andrecium, we can see the sepals here, and we can also see the petals here. We can also see the petals here. Now, how many of you understood the concept? Tell me in the chat right now. How the flower development actually happens on an entire plant. This is called as a flower. For a botanist point of view, for a biologist point of view, flower is nothing but a condensed shoot or a modified shoot, because the shoot here is undergoing modifications. How many of you understood? If you understood the concept, quickly some fire emoji in the chat to let me know, students. Yes sir, we are understanding. Sir, between stipule and axillary bud is a bud which, because it produces your branch or a stem. Stipules are the leaf-like structures. Leaf-like structure which main function is protection. Okay, they're two different things. How many of you understood? Quickly hit in the chat, students. How many of you understood the concept of what exactly is a flower? Yes? Okay. Third diagram. Understand, in third diagram what is happening here is this condensed shoot right here. This condensed shoot right here is getting modified. This condensed shoot is getting modified into thalamus, and on that thalamus the entire flower develops. Let me show you one more diagram. Okay. Let me tell you one more diagram, students. You'll get better understanding. Let's say here I have a here I have the thalamus. Here I have the thalamus. Okay. Here I have the thalamus part. On this thalamus, students, I will have leaf-like structure called as sepals. Can all of you see the sepals? Tell me students, can all of you see the sepals? Leaf-like structures called as sepals. Okay. Now on top of this, here in the middle, I have this structure. Can anyone tell me what is the structure in the middle? Anyone? What is structure in the middle? Okay. Now here I have one more structure called as your and then I have now students, a typical flower. This right here is a typical flower. Typical flower, right? Typical flower on the board. Now a typical flower has four main whorls, like whorls. Okay, the innermost whorl, right? So the outermost whorl. Right, let's go from outer to inside. The outermost whorl is made up of sepals. It is made up of sepals. Can anyone tell me what is this whorl called as? The whorl which is made up of sepal, it is called as calyx. It is called as your calyx. Calyx. It is called as calyx, the outermost whorl. Okay. Now students, then we have second whorl, which is made up of petal. Petal. What is this structure called as, students? Anyone? This right petal right here is nothing but the corolla. It is nothing but the corolla. Corolla. Then we have the male reproductive structure called as stamen. Stamen. And then in the middle right here, we have the entire carpel. Carpel. Now students, this stamen has multiple parts, carpel has multiple parts. For example, a stamen has this long filament and a anther. This entire stigma, or we can call it as a pistil here, or pistil or carpel here, right? Pistil or carpel has stigma, and this is the style, and this is the ovary. Ovary. What inside this ovary, students? Inside this ovary, we will have something called as ovules. So all the ovules will be found inside the ovary. Ovules. So all of you understand till here? Yes sir, we are understanding. Here is one more diagram I have given for you. So can you see calyx, corolla, and we have gynoecium. What is gynoecium, students? What is the difference between? Yes. Okay. So what is the difference between let's say gynoecium and pistil and carpel? Three terms. Listen to me very carefully. Gynoecium, pistil, carpel. Is everything same to same? Tell me yes or no in the chat. I will look at the chat right now. Tell me students, a simple question to all of you. Right. Is gynoecium, yes, good morning, JE aspirant also watching. Nice. Okay. Tell me, is pistil, carpel, gynoecium same to same? If yes, tell me yes sir. If no, say no sir. Okay. Yes, sexual reproduction is a general knowledge because everyone should know. Because many people do not know that a fruit which we get is coming from a flower. A flower is becoming a fruit. Very difficult to understand. Okay. Now tell me students, is carpel, pistil and gynoecium are same to same or are they different? If they are different, tell me what is different. If they are same, just say same sir. Yes. Okay. Let me tell you right now students, they are not same to same. All three are different actually. All three are different actually. Now let me tell you first the difference between a pistil and a carpel. Understand that first. Okay. Let's take this is my hand. My hand has multiple fingers. My hand has multiple fingers. Similarly, there is a structure called as pistil in the plant. A pistil will have multiple carpels. One carpel, second carpel, third carpel, fourth carpel, fifth carpel. So a pistil can have many carpels. Okay. If there is a single carpel, it is called as monocarpellary. If there are multiple carpels, multicarpellary. If all the carpels are free, apocarpus. If carpels are fused, syncarpus. So that is carpus. Pistil is the entire structure. When we have multiple pistils joined together, group of pistil is called as gynoecium. Similarly, let's say we have, what is the difference between stamen and androecium? Are they same? No. Stamen is individual. A group of stamen is called as androecium. A group of stamen is called as androecium. So all of these terminologies in botany, no one will teach you, because every teacher thinks that students, there are two things. Either the student knows everything or the students will not understand. Right? The reason teachers don't teach this is because they think two things. Either the student will not understand or the student already knows. So if either of two categories, right, I have taught you today. So if you understand the difference between a gynoecium, a pistil and a carpel, fire emoji in the chat right now. A group of carpels, many carpels joined together is pistil, right? Many carpels. We have many carpels, or a single carpel can also be there. A single carpel can be also there. Okay. It will give you the pistil. Multiple pistil will give you gynoecium. It will give you the entire gynoecium. So learn the difference. Learn the difference between the carpels, pistil and gynoecium. Very important. Okay. Now students, if this is out of the way, now here there is something called as essential whorls and non-essential whorls. Some of you might have heard about it in your older classes. Some of you might have learned it in your younger classes. Class 10th, class 11th, somewhere you might have learned there is something called as essential and non-essential. What is the difference? Understand the difference. Here, if you look at your entire flower, there are certain parts which are directly involved in reproduction. There are certain plants which help in reproduction secondarily. See, in the case of human reproduction I taught you, there is something called as primary sex organ, testis. Secondary sex organ is what? Ducts, glands. Secondary sex organs is indirect involvement. Same concept applies here. In the case of plant, there are certain sex organs which are, let's say, is non-essential. Non-essential whorls are the whorls which has, let's say, indirect role. It has something called as indirect role. Indirect role in reproduction. Example is going to be sepals and petals. Indirect role. Or we can say calyx and corolla. Calyx and corolla. Indirect role. Now, what could be the function of calyx and corolla? The main function of sepals or calyx is protecting the plant. What is the function of petals? The main function of petals is what, students? Tell me in the chat. Why do plants have such bright color petals? Many cases. To attract the pollinators. Then there is something called as essential whorls. Something called as essential whorl. This is basically the androecium and the gynoecium. Androecium and gynoecium. Why are they called as essential? Because they are directly involved in reproduction. Because they are directly involved. They are directly involved in reproduction. That's all. Because stamen can produce your gametes, that is your pollen grains, not the gametes, pollen grain, it can produce the male gametes later on. Okay. Now students, I hope that is clear to all of you. right now. Tell me yes or no in the chat quickly. All of you. Understanding botany students, all of you. Now let me teach you one more small concept. Is very simple. When you see on a plant, a flower is present on the plant. When you see a flower is present on the plant, the decision for the flowering, that yes, there has to be a flower, all that is taken long before. So the decision to make a flower is taken long before, and its entire thing is hormonal controlled. There is something called as floral primordia. This is the initial flower, not the flower. Floral primordia is the initial flower, which is not even present still. This floral primordia develops into floral bud. How come? Because of hormonal reactions. Because of hormones. Because of hormones, floral primordia is getting converted to floral bud. Now this floral bud will convert into flower. Now a question arises to all of you, right? This hormones, which hormones? You have studied in your plant growth. There are hormones like ethylene and auxin. Ethylene and auxin are the hormones. Auxin is the hormones which mainly responsible for flowering. Students, tell me, is every single chapter connected or not now? Tell me in the chat quickly. Does it make sense now? Does everything make sense? Okay, everything makes sense now? Hormone, we have floral primordia, convert to floral bud becomes flower eventually. This is the basic framework how the flower happens. Okay. Now students, let's go to the male reproductive part now. Shall we now? All of you? Tell me yes sir. All of you. Students, are you enjoying botany? Tell me yes or not. All of you are enjoying. Tell me students, yes or no? Because yes, sometimes it is important to learn NCERT lines, but it is also important to learn why the NCERT line is given there. Okay, why the NCERT line is given and how does it make sense to all of you. Okay, now students, I don't know why is my nose itching today. I think some insect bit my nose today. Stupid insect. Okay, now students, let's understand the stamen now. Stamen is the male part while carpel is the female part, which we all of us established. Establishment is done. Okay, now here a typical stamen, students, a typical stamen has how many parts? Let's say a typical stamen is there. This is the thalamus. This is the thalamus or receptacle. Okay, on this receptacle, students, let's say I have a stamen here. This entire thing is called as a stamen. Now this entire stamen has two main parts. Obviously all of you know that. This long slender structure is called as filament, and this round bilobed structure is called as anther. It is called as your anther. So anther and filament done. Till here very simple to understand. Now students, you need to understand here is that in this anther there are two locations. For example, this location here is called as distal end and this location is called as proximal end. Proximal end is the end where the filament is attaching to the thalamus or petals. We can see, right, petals are coming from here. So this is proximal end, distal end. Clear all of you? Clear students. Okay. Now, next concept to understand here is what is the meaning of bilobed and dithecous or tetragoniate? What is this microsporangium? What is this theca? Tell me how many of you have confusion in this following concepts. Raise your hands right now because I'm going to make it easy for you. Tell me in the chat. Okay. Students, all of you, quickly tell me in the chat. Hello, good morning, all of you. Hello macha to you. How does apple grow? That is false fruit. I'll tell you later on. Don't worry. Okay. Now, how many of you get confused between to understand what is bilobed, what is dithecous, what is tetragoniate, or what is microsporangia, where is sporogenous tissue, all that concepts? Raise your hands quickly, all of you. Quickly. All of you raise your hands. Quickly raise your hands. Don't be shy. Do not be shy. You can raise your hands. I'm going to make it easy for you to understand. All of you drink some water. Quickly drink some water and we'll go ahead. Yeah, good morning. Oh, how many times good morning today? Heavy good morning today. Good morning to all. Let's finish this chapter. Macha means it's a Tamil word which means brother-in-law, but in Bangalore it has become like how you refer to as a bro, right? We always say bro. Okay. Now listen to me all of you quickly. Now let's say this is your entire anther, filament as well, this entire stamen, filament and anther. Here can you see there is a longitudinal groove here. Okay, runs separating theca. Now what is this theca? What is longitudinal groove? What is the lobe? Understand very carefully now. Understand very carefully. Okay. Listen to me. Let's say this is an anther. This is an anther right here. I take this anther and I chop it in half. Right. I see an anther and I chop it in half then and there. And I look at the anther from the top view. Let's say I take an anther and I chop it off and I look at this anther from the top view like this. What do I notice? What do I notice is this structure right here. This structure right here. Now listen to me all of you very carefully now all of you. This is a structure of a typical anther. Not all anthers are the same because there is a family called as Malvaceae where difference can be there. But this is an example given your NCERT. Okay. Now if you look at that anther from the top view, it has two lobes. This entire thing on this side is called as lobe 1. Lobe 1. This entire thing on this side is called as lobe 2. Let's take, this is the anther and I open it up. Tell me how many lobe do you see, students? This entire thing is one lobe. This entire thing is other lobe. So how many lobes are there? There are two lobes here. There are two lobes. So can I call it, students? Can I call it as bilobed structure now? Tell me yes or no. Students, quickly in the chat. Can I call it as bilobed structure? It is called as bilobed. So lobe 1, lobe 2, bilobed structure. Now imagine, all of you imagine, okay. Now in this entire lobe, I have two small holes here. This one lobe has two holes. This lobe here, one hole here, one hole here. So this hole or a cavity, all of you know what is a cavity, right? Gap or a hole. It is called as a theca. So there are 2 thecas in one lobe, 2 thecas in one more lobe. So there are students, how many tell me? One lobe has how many theca, students? Theca 1, theca 2. So can I call it as dithecous condition? Dithecous for one lobe. This is for one lobe. Now if there are 2 thecas in one lobe, tell me students, how many thecas are present in total here? How many thecas are present? 4. So can I also call it as tetrathecous condition? Yes. Tetrathecous condition is only present. Tetrathecous is only present when I consider 2 lobes. Clear all of you? Clear? And between the 2 lobes, there is a connecting link here which runs longitudinally. That is called as connective in the right in the middle here. This is mainly made up of parenchyma. Parenchyma. So one lobe, 2 lobe, bilobed. One lobe has 2 theca, dithecous condition. Now students, do you think this theca or the cavity is empty or does it have something? Anyone? This entire theca, do you think it's empty cavity or there is something is present there? Something is like present there. Something is present there. Anyone? This theca, students, is actually not empty. These cavities are not empty. These cavities, students, has something called as microsporangia. So there is microsporangia present here. Microsporangia present here. Microsporangia is present here. The other name of this microsporangia, it is also called as pollen sac. It is also called as pollen sac. So please remember this. If someone asks you in examination, there's no pollen grains. No. Pollen grains are not present here. Pollen grains will be only present in mature anther. This is immature. So immature anther will have microsporangia, also called as pollen sac. It is a sac. Inside this sac, there will be pollen grains later on. So here we have microsporangia. Now students, how many theca do we find here? I told you 4 theca. 4 theca are present here. And 1 theca has 1 microsporangia. So can anyone tell me how many microsporangia do we have in total? 4. So if there are 4 microsporangia in total, can I also call it as tetrasporangiate structure? Tell me yes or no. Tetrasporangiate structure? Yes, sir. Or in NCERT, it also calls tetragonal. Tetragonal is also mentioned in NCERT. So tetrasporangiate because there are 4 microsporangia. Bilobed condition because there are 2 lobes. Dithecous because every single lobe has 2 theca, or tetrathecous because of 4 thecas here. Telling me how many of you actually understood without lying all of these following concepts? Raise your hands. Raise your hands. Or just say yes sir, we understood the entire concept today. All of you quickly in the chat, and also smash the like button. All of you quickly smash the like button if you understanding all these concepts. Tetrasporangiate, 4 microsporangia, bilobed, dithecous, tetrathecous. Or some fire emoji in the chat also makes sense. Fire emoji in the chat also makes sense to me, because yes, you understood. Because I'm going to have to look at the chat here on the phone. All of you understood? Yes? Clear? Beautiful. Beautiful. Okay. Now students, let's go ahead. Now, this is your anther, this is your pollen sac. Can you see pollen sac is also called as your microsporangium. This is called as line of dehiscence, where the dehiscence happens, and pollen grains are present here only in mature anther. Only in the case of mature anther, not before. Okay. Now students, now we understand microsporangium. We learned about microsporangia there. We learned about microsporangia, right? Yes, we learned. Now this microsporangia has 4 main layers or 4 main walls. Can all of you see this diagram? We do this diagram, right? Same diagram was there, right? Here can you see this one, each one is called as what? Each one is called as microsporangia. Microsporangia or microsporangium here because of 1. So this microsporangium has 1, 2, 3, 4. 4 different layers here. Okay. If you can notice here students, the outermost layer is called as epidermis. Epidermis is the outermost layer. Then we have something called as endothecium, that is the second layer from the outside. Middle layer is the third one, and the innermost layer is called as tapetum, is the innermost layer. Now what will they ask you in examination? Will they ask you the layers? There is a chance they can ask you the different layers. But the main thing which they can ask you is the functions of each one of them. And for the very same reason. Okay. Before I show you the functions, let me give a small understanding of what we have right now. Okay. This entire structure is called as what? This entire structure is called as microsporangium. Okay. It has how many layers? Students, how many layers are there? 4 layers are there. Epidermis, endothecium, middle layer, and tapetum. But what is present inside the microsporangium now? They're going deeper and deeper. So there is a tissue found inside microsporangium, that is called as sporogenous tissue. Sporogenous tissue, students, sporogenous tissue. So what do we understand right now? First, we understood there is something called as anther. We understood there is something called as stamen. First of all, stamen. A stamen will have something called as anther. A anther will have something called as a lobe. A lobe will have something called as theca. Inside theca we will find something called as microsporangium. Now inside microsporangium, the innermost thing inside microsporangium, we will find sporogenous cells. This is your understanding till now. This is your flow chart till now, students. Stamen, anther, lobe. Inside lobe we have a theca. Inside the theca we have microsporangium. Microsporangium has multiple layers, and the innermost layer is called as sporogenous cells. And these sporogenous cells, students, these sporogenous cells, this right here, will develop into pollen grains. How many of you understand? Tell me yes or no in the chat. Quickly, all of you. I'll give you exactly 10 seconds to absorb all the information present on the board. Right. Absorb all the information and tell me, sir, yes, we are following the flow chart. How many of you following? So stamen has anther, anther has lobe, lobe has a theca, theca has microsporangium. That microsporangium has in the inside it has a sporogenous cells. Sporogenous cells can give rise to the pollen grains eventually. Is the flow chart clear to all of you? Science is all about flow charts. If you understand the flow chart, you're good to go. Yes? All of you? Understood? Nice. Nice. Student, this is so much fun, right? See, look at this. It's so easy to understand. So easy to understand. Because if you don't understand the flow chart, right, there's a very high chance that you get confused. Sir, what is sporogenous? What is microspore? Microsporangium? What is microspore? All that confusion is clear now. Okay. Zero level very difficult, bro. Zero level very difficult. Zero level. Because there's so much of syllabus to cover. You should have some background knowledge to clear right now. I will not lie and tell you that, you know, 0 to 700 in 1 month, 2 months, not possible, not possible. Unless you have photographic memory, unless you know every single concept in the world, you're revising, then yes. Okay. Now, again, epidermis, endothecium, middle layer, tapetum, sporogenous cells. This is a microsporangium. Microsporangium. Okay. Now here are the functions I've given for all of you. Functions is very easy to understand. Can you see here? Epidermis is what? Outermost layer, single layer whose function is to protect the pollen sac. Protection. Endothecium. It is present inner to the epidermis. Inside epidermis it is present. Okay. Here we have something called as alpha cellulosic fibrous bands. This one is not required for you. This one is not required for you. The only thing which you need to remember for endothecium, the function of endothecium, endothecium helps in dehiscence to spread the pollen grains, helps in release of pollen grains. And this endothecium right here, students, it is very short-lived. It is very short-lived. Ephemeral we say, short-lived. Then we have the middle layer. It is present inner to endodermis. Oh, this is also short-lived. This is also short-lived, students. This is also short-lived. Usually endothecium can be still there, but compared to endothecium, middle layer is more short-lived. Okay. Degenerates at maturity. The innermost three, the first three, main function of first three is protection. Protection. Protection of the pollen sac. Apart from protection, they can also help in dehiscence, that is the release of pollen grains. But the innermost layer, okay, innermost layer, the main function of innermost layer is it provides nutrition. It provides nutrition. Okay. Right here, students. It provides nutrition. Nutrition. Okay. Now, apart from nutrition, students, the main point we should remember here is, apart from nutrition, is that tapetum cells possess dense cytoplasm. That point is important. Okay. They are also multinucleated condition, multinucleated condition, or polyploidy can be seen here. Okay, polyploidy can be seen here. Apart from that, tapetum also produces an enzyme called as 𝐂𝐤 enzyme, which I'll tell you later on. 𝐂𝐤 enzyme. Apart from it, also produces something called as pollen kit. Pollen kit. Pollen kit. It is produced by tapetum itself. Tapetum only is producing everything. Pollen kit also. Pollen kit is a sticky substance which helps in attracting the pollinators. Pollen kit. Okay. Apart from this, tapetum is also responsible for production of sporopollenin. Also produces sporopollenin, students. Sporopollenin. Also produces sporopollenin, students. Sporopollenin. Okay. So that is your main functions of your tapetum. Main functions of epidermis, endothecium, middle layer is protection. Main function of tapetum is to produce sporopollen, nutrition, 𝐂𝐤 enzyme, and pollen kit. Will all of you remember this or not? Tell me. Will all of you remember this? Tell me yes or no. Tapetum functions. All of you remember this? All of these notes will be available on Telegram channel today itself. Okay. Or in the description of today's video, you will find these notes. Download these notes. Use these notes for your learning. Easy go. Okay. Clear all of you? Clear? Tell me yes or no in the chat quickly. Yes or no? Quickly all of you. See, you might have heard about only nutrition before. This is extra information for all of you. Sporopollen, pollen kit and 𝐂𝐤 enzyme. All of you remember? Okay, very good. If you can remember that, now I will teach you something called as microsporogenesis in the most easiest way possible. Ready all of you? Ready all of you? Yes? Quickly in the chat all of you. Ready? Microsporogenesis is very important concept. Many students fail to understand this because you by heart all this. Okay. Now, where does it start? Microsporogenesis, where does it start? Okay. I'll give you a picture. Try to imagine. We have the entire flower. Our entire flower has something called as your stamen. A stamen has something called as anther. A anther has something called as lobe. A lobe has something called as 𝐤𝐤. Here, this flowchart again. Lobe has something called as 𝐤𝐤. A 𝐤𝐤 has something called as microsporangium. A microsporangium has something called as sporogenous cells, and this sporogenous cells right here, these sporogenous cells, this is where the microsporogenesis start. Many of the sporogenous cells, they develop, they increase in size, they become large, and it gives rise to a structure called as MMC. It gives a structure called as MMC. What is MMC? MMC is nothing but megaspore mother cell. Megaspore mother cell is called as MMC. And this MMC, remember students, it is from sporogenous cells which is diploid. This MMC is also going to be diploid in nature. MMC. Now this MMC, students, undergoes a process called as reduction division. Meiosis 1. First meiosis 1. After meiosis 1, what happens, students? We get two cells. These two cells, students, can anyone tell me, are they haploid diploid now? Obviously, after meiosis 1, they become haploid in nature. Now they undergo second division. That is called as meiosis 2. And we end up getting 4 cells now. We end up getting 4 cells. We end up getting 4 cells here. The thing to be noted here, students, that all of these 4 cells, right? All of these 4 cells are completely joined together. All of these 4 cells are completely joined together. These 4 cells is nothing but they are called as microspore tetrad. They are called as microspore tetrad. This is called a microspore tetrad. Students, please tell me in the chat, are they haploid or diploid? This microspore tetrad, are they haploid or diploid in nature? All of these are haploid in nature. Now, all of these microspore tetrad, students, they will eventually become free. All of these microspore tetrad, students, they will eventually become free. From each other. How is that possible, students? It is possible because of the enzyme 𝐂𝐤 enzyme. Now, can anyone tell me, students, who is producing this 𝐂𝐤 enzyme? This 𝐂𝐤 enzyme is coming from where? This 𝐂𝐤 enzyme is coming from the tapetum. Clear? Now, this right here is the microspores. This right here is the microspores. Microspores. Okay. Now this is called as your simultaneous type. It is called as simultaneous type of division. Why? Because here we have meiosis 1, meiosis 2, and then we have wall formation. This is simultaneous type. Okay. Now there is something called as your simultaneous is there, and then there is continuous type. Okay. In successive type, what happens is we have in successive type, what do we have, students? We have meiosis 1, cytokinesis, meiosis 2. Okay, and then again cytokinesis. This is called as simultaneous type. Meiosis 1, cytokinesis, meiosis 2, cytokinesis. Successive type. The one I taught you something called as we have meiosis 1, meiosis 2, and then cytokinesis. Okay, this is mainly happening in the case of your monocot. This happens in the case of dicot. This is not given your NCERT. I'm just telling you, you might not be aware of this. So if you want to remember this extra information, you can remember. Okay. Now, I just realized. Then fixed. So what happens is we have microspore mother cell, meiosis 1, 2 cells, meiosis 2, 4 cells, and then we have microspores. Okay. Then we have microspores. Okay. Now students, is this the end of it? Students, is this the end of it? Does the entire thing end here? But once we get the microspores become pollen grains? No. There is actually next step called as formation of pollen grain. What exactly happens in the formation of pollen grains? Let's take a microspore. From there. Let's take a microspore. This microspore is haplodiploid. It is haploid, obviously. Microspore is haploid in nature. Now this microspore, students, undergoes a reaction where in this entire microspore, students, there will be a large vacuole formation. Okay. Vacuole formation is happening. When this large vacuole formation is happening, the entire nucleus is sent to the corner. Okay. Now what happens? Understand. Now this entire nucleus now undergoes division. Okay. Which type of division, students? It undergoes a division which we always call it as equational division. What is that? That is called as a mitosis. It undergoes something called as mitosis now. But this mitosis, students, is not very simple. This mitosis is actually asymmetrical. Okay, it's called as asymmetrical mitosis. It is called as your asymmetrical mitosis because the spindle fiber formation happens here very weirdly. This is your vacuole. Here there will be a spindle fiber formation because obviously if mitosis has to happen, there will be spindle fiber formation has to happen because spindle fibers will pull apart and the entire chromatids will separate out. Okay. Now this will eventually result in asymmetrical mitosis will eventually result in the male gametophyte. Okay. This male gametophyte will have two nucleuses. This male gametophyte will have an outer layer like this, and this male gametophyte is nothing but the pollen grain. This is nothing but the pollen grain. Okay. So students, that's how we end up getting the pollen grain. And this two nuclei here, one is called as your vegetative, other one is called as generative cell, vegetative cell. Because there is a crossal formation here, now they become cell eventually. Here you can see this is much more larger, this is much more smaller here. This one has more irregular shape, large abundant reserve food material. This one is very small, and this generative cell, students, eventually will divide, undergoes mitosis again, and give rise to two more male gametes eventually. Okay, so I'll give you a small recap what we learned right now. We learned that the entire microsporogenesis starts where? The entire microsporogenesis starts at the sporogenous cells. Remember that point. Sporogenous cells, one of them become very large and they give microspore mother cell MMC. I wrote megaspore even become, that happens in the case of megasporogenesis. Okay, it undergoes meiosis 1, 2 cells, meiosis 2, 4 cell. This is called microspore tetrad. This microspore tetrad eventually undergoes freeing up, that is because of 𝐂𝐤 enzyme. Okay, now this microspore, let's take one microspore, it undergoes vacuole formation. There's a formation of vacuole here. Can you see large vacuole formation? It's called vacuolization also. Okay, then after vacuolization it undergoes mitosis, asymmetrical mitosis or asymmetrical spindle fiber formation. Okay, and that will produce something like the structure, asymmetrical spindle fiber, and eventually we get a structure called as your male gametophyte or pollen grain, which has vegetative cell and generative cell. How many of you understood? Raise your hands quickly. How many of you understood? How many of you understood the entire structure? If you can see your NCI diagram also tells you this. If you look at your NCI diagram, you can see here. This is right here is what, students? This here is a microspore. Microspore is undergoing vacuole formation. Can you see all of you here NCI diagram? It undergoes vacuoles formation. Can you see large vacuole here? Large vacuole. Now, eventually what happens? Asymmetrical spindle formation happens. What happens later on? Later on, this cell divides into two cells, that is by mitosis. Mitosis completely, giving rise to vegetative cell and generative cell eventually. It is there in your NCERT also, what I just told you. That is called as your pollen grain formation or male gametophyte formation. Why male gametophyte? Because this generative cell will give the male gamete later on. Pollen grain is gametophyte. How many of you understood this entire structure? Look at this, NCERT makes it so easy to understand here. But students, tell me, unless you understand this entire flow chart, tell me, this becomes difficult to understand. Tell me yes or no. Tell me yes or no. It becomes difficult to understand. It does. So your job as my teacher is to make you understand NCERT is easy only if you look at it right angle. Left angle, if you see it, not proper. Okay, right angle you should see it. Okay, then NCERT becomes easy. Okay, nice. Now, if you can notice here, students, here this entire microsporangia or microsporangium is not present at all. Why? Because entire microsporangium is degenerating, and can you see pollen grains are coming out in that region? Because here we had microsporangia which had the sporogenous cells. Multiple sporogenous cells got converted to pollen grains. Nice? Tell me, nice or not? Nice. Okay. So pollen grains represent the male gametophyte. A pollen grain is a male gametophyte. Okay. It comes in many shapes and sizes. Okay. Now, here is a very important thing, students, the structure of pollen grain. If you look at the structure of pollen grain which we have right now, so precious. Now, all of you know how difficult it is to form a pollen grain. Now, finally, we have the precious pollen grain. Let's try to find its shape and size. Now, what is present in pollen grain? Okay. Now, if you look at a pollen grain, students, it has two main layers. Okay. Before we go to the layers, let me tell you the cells inside. Students, can all of you see? This is a large cell. This is called as vegetative cell. and the small one is called as generative cell. Difference you understand. This vegetative cell, student, vegetative cell, right? It is large, irregular shaped nucleus. Tell me yes or no. If you can go back here and see, tell me, large irregular shaped nucleus. Yes, obviously. We don't draw anything randomly. Large irregular shaped nucleus is present here. And this particular structure, students, it has something called as abundant food reserve. It can be present here. And this vegetative cell, it has a separate name. It has one more name. It is also called as tube cell. Now you might ask me, sir, why do we call it tube cell? What is the necessity to call it tube cell? Because during the pollen tube formation, the cell which is leading the entire adventure, imagine there is a long tube and you don't know where to go. The entire guide of your male gametes is done by a tube cell. This is the tube cell which will sacrifice itself to make sure that this generative cell reaches the target. So this is like this tube cell is the sacrifice which we give during fertilization. Okay. Now this is called generative cell, student. Generative cell is very small. It is very small and it floats in the cytoplasm of generative cell, students. And this generative cell itself, will divide eventually and it will give rise to two male gametes into two male gametes eventually. This generative cell right here. Okay. Now apart from the students, apart from the cells here, this entire pollen grain has an outer layer and inner layer. The outer layer here is called as 𝐂𝐡𝐞, and this 𝐂𝐂𝐡 is very tough. 𝐂𝐂𝐡 is very resistant to microbial action, it resistant to biological activities. Why? Because this entire 𝐂𝐂𝐡 is made up of a chemical called as sporopollen. It is written here somewhere here. It is made up of sporopollen. Now a question to all of you, my students. This sporopollen is produced by which layer of Microsporangium? Anyone? This sporopollen which is present in the 𝐂𝐂𝐡, where do we get it from? Sporopollen. This sporopollen is obtained from tapetum. Itself. Okay. Now, next layer. Now, if you look at the structure of your entire pollen grain, right? This is the pollen grain, and here is the 𝐂𝐡𝐼. Now students, in this entire 𝐂𝐡𝐼 layer, right, there is sporopollen. I told you sporopollen is there, right? Now in this entire structure there are small gaps here where the sporopollen is absent. In this gap, sporopollen is absent here. This entire structure is called as germ pore. This entire structure is called as germ pore, where the sporopollen is absent here. Now students, now all of you think and tell me. Let's say this is intine. I want all of you to understand the concept today. This is intine. This is 𝐂𝐡𝐼. What exactly happens during fertilization? Let me tell you right now. Listen to this concept very detailed. It's very difficult. No one usually tells this. I don't know why. Okay. During fertilization, what happens is this 𝐂𝐡𝐼 is not present here, right? This entire pollen grain will absorb the moisture present on the stigma. This entire pollen grain will absorb the moisture from the stigma, and the pollen grain will expand. The pollen grain is expanding through this germ pore. through this germ pore, the intine is coming out actually. Through this germ pore, the intine is coming out. And this is the pollen tube which you study in your entire double fertilization. So the intine, can you imagine? All of you have to imagine. This entire intine is getting pressed outside and intine is coming out. Okay. Pollen tube arises by extension. Let me write here. Pollen tube arises by extension of intine. The intine gets extended here. The entire intine is getting extended out as a pollen tube. All of you are able to visualize in your mind, tell me yes or no. All of you are able to understand this concept? How the intine comes to exist, how the pollen tube comes into existence? The entire germ pore, through the germ pore, the intine is squeezed outside and we get the entire pollen tube. Okay. This is a germ pore. Germ pore, done. Okay. Now, inner layer is called as intine. Intine is little elastic. Intine is more elastic and intine is very soft because it is made up of cellulose and pectin. It is made up of cellulose and pectin. Cellulose and pectin. That is your entire intine. So exine is made up of sporopollen. There are small gaps called as germ pore. The intine is made up of cellulose and pectin. Okay, done. Now a question to all of you. Okay, students, tell me in the chat quickly. Do you want to learn one advanced concept or not? Or do you want to stick to NCERT only and make your life easy? Okay, even if you say no, I will tell you, you know that, right? Right. Usually what happens, what do you think, students? On a pollen grain, do you think there are a single germ pore or do you find multiple germ pore? Tell me in the chat. Students, we spoke about germ pore here and we saw pollen tube development. Do you think there is only a single germ pore or multiple germ pore, students? So there is a very high chance, right? There is a very high chance that there can be a single germ pore, that is in the case of, that's called monocolpate, and that is present in the case of monocots. But if there are 2 germ pore, 3 germ pore, that is called as bicolpate and tricolpate, and that is present in the case of dicots. So learn this difference. See, what we teach you at your class 12th level about monocot and dicot is very basic. We always teach you, like, single cotyledon, 2 cotyledon, scutellum is there, yeah, albuminous, exalbuminous. But there are multiple differences. As you go higher and higher in learning botany, you will understand the difference even in the pollen grain. Okay, difference in the pollen grain. Okay, right? So monocolpate, bicolpate. Okay, so if you from my class almost today, you will know the difference now. Extra information in your brain stored today. Okay. Now students, now all of you know vegetative cell, generative cell. Vegetative cell and generative cell. Okay. Now, how many cells do you see, students? Can anyone tell me? Do you see 1 cell or 2 cell here? This is called as 2-celled stage. It is called as 2-celled stage. 2-celled stage. Now this is the stage where 60% of angiosperms will shed the pollen grain. 60% of angiosperms will shed the pollen grains at this stage right here. This stage right here. What happens in remaining? In remaining 40%. In remaining 40% of your angiosperms, what happens? This entire cell here, right? Here we have, let's say, your vegetative cell. Here we have generative cell. This generative cell, students, will divide now. This generative cell will divide into two more cells. Can you see here? This generative cell will divide into two more cells, will give rise to two male gametes in the pollen grain stage itself. And this is called as three-celled stage. Three-celled stage. Three-celled stage where division directly happens. And this is the pollen grain in many cases. In 40% of angiosperms, this pollen grain is released. In 60%, this is released. So know the difference. Okay. Now pollen grains can cause allergies. I am very susceptible. I do not like pollen grains, students. I do not like pollen grains. Okay. To eat, probably I will eat it. I will try eating it. But if I take a pollen grain, achi starts. Okay. That's called pollen allergy. Because pollen grains can get stuck in your nostril and it can cause havoc there. Okay. Major issue. Okay. So it can cause respiratory disorders, asthma, bronchitis also. There are many diseases which can be caused by your pollen grain. There is a famous example is Parthenium. Parthenium is also got carrot grass because the pollen grains are easily shed here, and these pollen grains can cause many of the respiratory disorders. Okay, respiratory disorders. Stupid pollen grain. But pollen grains are used also. There's a use for pollen grain. The use of pollen grain is what it can be eaten. It can be eaten by athletes or it can be eaten by horses for the immediate energy. Why? Because the pollen grain has a lot of nutrition. It has a lot of nutrition. The tapetum, remember the tapetum was there? No, the tapetum is providing all that nutrition. Okay. So pollen grain has a lot of nutrition. Now students, if that concept is clear to all of you, there is something called as pollen viability. What is pollen viability? The meaning of pollen viability is that once the pollen grain is released, once the pollen grain has been released, how long it can stay active before fertilization or before landing on the stigma, it is active, like hyperactive. Now this pollen viability is very variable because in some cereals it is only for 30 minutes, but in other cases for months together it can be there. Let me tell you, let me show you NCERT lines. Okay. See here, such as cereals such as rice and wheat, pollen grains can lose viability within 30 minutes. Maximum is 30 minutes. After that, they are completely not useful. But in some members of Rosaceae, Leguminaceae, and Solanaceae, these three family, they can be viable for months together. Or what if I tell you a pollen grain can be viable for hundreds of years? Will you believe me? Students? What if I tell you a pollen grain can be viable for hundreds of years? Yes, it can be. How? Because of liquid nitrogen. They can be viable for hundreds of years. Okay. Species of for years in liquid nitrogen. Okay. So there is something called as pollen bank. We study this concept of pollen bank in your ex situ conservation also. We have seed bank, pollen bank. Okay, for hundreds of years pollen grains can be viable. Okay, be viable. And students, with that your entire stamen in detail is done now. Let me give a small 2 minutes recap what we learned till now before we go ahead, before you don't forget everything. We started with the flower. I told you there is something called as, what is a flower? Condensed shoot. A flower, this is a typical flower. It has 4 whorls: calyx, corolla, androecium, gynoecium. Done. Then we learnt about your non-essential, essential whorls. Yes, done. Then we learnt about a flower has to develop, it has to undergo, floral primordia has to undergo hormones production. Then we get a flower. Then we learned about stamen. We learned about the structure of a stamen, filament and anther. Then we learned something called as anther structure. We learned about the bilobe, dithecous, tetrathecous, tetrasporangiate structure. Right here. Tetrasporangiate. Then we learned about your Microsporangium 4 walls, epidermis, endothecium, middle layer, and tapetum. And inside the entire Microsporangium, we have something called as sporogenous cells. These sporogenous cells will give the pollen grains eventually. Okay, eventually. These are the different functions of each of the layers. We learned that tapetum is multinucleated. It is dense cytoplasm. It can provide nutrition, sporopollen, callase enzyme, pollen kit. 3 layers, protection main function, and dehiscence. Dehiscence means release of pollen grains. Microsporogenesis, sporogenous cells, MMC, microspore mother cell undergoes meiosis 1, meiosis 2, callase enzyme, microspore tetrada free. Okay. Then we learned about your pollen grain formation. Microspore undergoes vacuole formation, large vacuole, nucleus undergoes mitosis, asymmetrical spindle fiber, and we end up getting a pollen grain. Okay. Here is the information more. Pollen grain can be of different shapes and sizes. Pollen grain has 2 main cells, vegetative, generative. Vegetative is dense, large, irregular shaped nucleus, dense cytoplasm. Here, generative cell is very small. This generative cell will float. It will float in the cytoplasm of vegetative. Okay. This can give rise to 2 male gametes eventually. Here we have something called as 𝐂𝐡𝐼ne and 𝐿𝐻𝐼ne. 𝐂𝐻𝐼ne is made up of sporopollen. 𝐿𝐻𝐼ne is made up of cellulose and pectin. Here is something called as germ pore. Through germ pore, 𝐿𝐻𝐼ne is coming out in the form of pollen tube. 2 cell stage, 3 cell stage. Pollen allergy, pollen tablets, pollen viability. And that is the entire in detail explanation of your entire stamen. Did all of you enjoy the entire stamen? Tell me yes or no. Did all of you enjoy the entire stamen full detail explanation? Microsporogenesis, entire thing. Because even in Megasporogenesis, I'll teach you something which you do not know till now. Even in Megasporogenesis, I will teach you concepts like which you have never learned before, and it will make so much of sense. Your NCERT will make more sense. Okay. So all of you, can we now start the entire Gynoecium? Can we start? All of you. Let me see the chat properly. All of you quickly smash the like button. Students, share the video. What is this? Share the video, friends. There are very less live watching. What is this? Such detailed explanation you will never get. Very rarely. Okay. And probably the last time I'll be doing this chapter. Probably the last time I will be ever doing this chapter, and probably this video will also get deleted eventually. Because this is Gopika Ma'am's lecture. This is Gopika Ma'am's class. No. Right. This is Gopika Ma'am's chapter. Right. So probably within 2 weeks this video also will get deleted. Okay. Don't worry. So, or this end of this month. Video will be only there till the end of this month. After that it will get deleted. Short time video only. Short time class. Okay. So try to learn today only as much as possible. Get the notes. Make your own notes from this video today only. Okay. Okay. So like the video. Let me know in the comment section how was the session. Okay. So I will have very less time to even see the comments also. Okay. Now let's go to the next concept that's called as Gynoecium. Questions are in the end. Okay. Now students, Gynoecium, right? Gynoecium or the carpel here, group of main unit here, carpel, it has 3 main parts. Okay. There is something called a stigma, style and ovary. Now students, how many of you get confused in understanding what is a locule, what is a placenta and what is the ovule? Raise your hands. Quickly raise your hands. How many of you get confused between what is a locule, right? What is a locule, what is a placenta and what is a ovule and what is ovary? Is there a difference or all of this same to same? Tell me in the chat quickly, all of you. Do you think all of them are similar, or all of them are different to each other? All of you quickly in the chat, tell me. Is ovule, ovary, placenta, locule, is everything same? No, it's not. Let me tell you exactly right now. See, this entire structure is right here, is called as ovary. It's called as ovary, is the outer part. Now, inside the ovary, what do we have? Inside the ovary, this I'll tell later on, students, wait. Main concept is coming. Now, if you look at inside the ovary, this is a structure which you get. Now, this structure, if you see here, inside a ovary, there can be many ovules. Can you see? These are called as the ovules. And here, there is a chamber. This entire cavity here is called as locule. Let me show you one more diagram. All of you like this? Students, all of you know the structure. What is the structure? This is a lady's finger. Here we can see axial placentation, we can see here. Okay. Now can you see, students, here is a chamber here. If you, the seed is nothing but ovule. This seed was before ovule only, right? Now imagine there is an ovule here and there's a cavity. Can you see all of you gap here? This entire cavity is called as locule. It is called as locule. The hole will be in a hole. The hole is called as locule. Okay. And this entire structure here, this middle part, in the ovary, that is called as placenta. That is called as placenta. So there is placenta here and all the ovules are attached to the placenta. Can you see? All the ovules are attached to the placenta. Tell me yes or no. If you can see in this diagram also, all this is the placenta here right in the middle, placenta. All the ovules are attached to the placenta like this. All the ovules are attached to the placenta like this. Okay. Now students, this is the stalk. Let me draw it for you here one more time. Okay. Let's say this is the placenta here and there's a stalk like this and we have ovule here. Let's take one more stalk here. We have ovule here. Ovule here. This right here is the ovule. Okay. And this ovule is present in a chamber. The chamber is called as locule. Locule. Now students, what do we call this stalk here? Can anyone tell me? This ovule is attached to the placenta. This entire ovule is attached to the placenta here. Middle part is the placenta. Okay. This entire ovule is attached to placenta by a stalk. What do you call that stalk, students? Anyone? Anyone, students? This is the placenta here. All the placenta is attached in the middle and ovules are attached to the placenta by help of a small stalk. Anyone? What do you call that stalk as? Anyone who can tell me what is the stalk called as? Anyone? Quickly tell me in the chat. What do you call that stalk as? You have learned it before. You have learned it before. Anyone who can tell me? I'll give you exactly 10 seconds to think. 10 seconds to give it a nice think. And once I tell you, you will get like, yes sir, it makes sense now. Botany is so easy, you will tell me. Students, when you look at this is the anatropous ovule structure. Here we have this structure called as funicle. How many of you know what is a funicle? Imagine this funicle is attached here. This is the funicle which is attaching here, and then we have this ovule. This entire ovule is present right here like this. Does it make sense to all of you now? Not hilum, not hilum, not hilum. I'll tell you what is hilum now. So this is called as funicle. Funicle is a stalk here, and this stalk is attached to the placenta. So arising from the placenta are the microsporangia, commonly called as the ovule. Ovule is other name is megasporangia. The ovule is small structure attached to the placenta. Ovule is attached to the placenta by a stalk that is called as funicle. How many of you understood concept? Because it's very easy to by heart all this. I'm telling you again, very easy to by heart all of this thing. I'll give you this notes, you will read this and you'll by heart it. But I want you to visually represent in your mind and understand. So this is the stalk here, this is the ovule, it is attached like this, and this is the funicle here, okay. Now let's say this is the funicle here, this is the entire ovule. At one point this funicle and this ovule, they're attaching here, right? Students, tell me, this funicle here and this ovule is here. At one point they're attaching this point here. What do you call this point of attachment? Anyone? What do you call this point of attachment where ovule and funicle both are attaching to each other? Okay. What do you call that? All of you. Quickly. If you see here also, here we have funicle, the stalk. Here we have the ovule. Both of them are attaching at this point here, X mark. Both are attaching at this X mark here. I can show you one more diagram. See here. This is the funicle. This is the ovule. The point of attachment, the body of the ovule fuse with the funicle. Ovule and funicle both are fusing. Region is called as hilum. That is called as hilum. So hilum is the point of attachment between the funicle as well as the ovule. Okay, beautiful. And this entire ovule has outer layers. The outer layers are called as integuments. Integuments are the outer layers. Outer integument, inner integument. And students, remember, these are the integuments which will become seed coat eventually. Seed coat. They will become seed coat eventually. Integuments. Okay. Now, here this entire integuments are coming from top to bottom. But at one point, integuments are not joining. The point where integuments do not join, that point is called as micropyle end, where integuments are not joining. Can all of you see this? Integuments don't join. And the opposite side is called as chalazal end. And inside this entire megasporangium or megasporangium. I thought I said micro again. In this entire megasporangium, there is a small structure in the middle. Who can tell me what is that? That is called as female gametophyte. Anyone? The structure present in the middle is called as embryo sac. Embryo sac right in the middle. Embryo sac. So inside the ovule we have the embryo sac, also called as the female gametophyte. Female gametophyte. Similar to pollen grain. Similar to pollen grain, here we have female gametophyte. Around the female gametophyte, we have multiple diploid cells called as nucellus. Multiple nucellus cells. Nucellus. And there are so many types of ovules where NCR tells you this one, an anthropous ovule. There is something called anthotropous, amphitropous, campylotropous, hemianthropous. Right? There are multiple types are there. Now students, can I help you revise the entire ovule in 7 points now? Can I help you to revise the entire ovule in 7 points? Tell me yes or no. 7 points theory. Or tala for a reason, okay? Can I teach you now? Just 7 points. Understand the following entire ovule now. Can we go? Can we start? 7 points. The first point is what? The first point is funicle. Funicle is the stalk of the ovule. Done. First point. Second point. Understand. Second point is called as hilum. Hilum is the point of attachment for funicle and the ovule. That is called as hilum. Third point is the integuments. Outer and inner integument. Now, fourth point is micropylar part, where the integuments do not join. Fifth point is chalazal end, the opposite side of micropylar. Okay. Sixth one is the nucellus. Nucellus is the all different type of cells here. Mass of cells called as nucellus, diploid cells called as nucellus. Sixth point. Now, in this mass of cells, there is something called as your entire female gametophyte. That is the embryo sac. 7 points. So in the entire 7 points, you are able to revise the entire embryo sac, funicle, hilum, integuments, micropylar, chalazal, and the embryo sac. Tell me, isn't this topic so easy to understand? Tell me honestly, how easy it is to understand the entire topic of your megasporangium or the ovule, right? And this is a funicle which attaches the placenta in the entire ovary. Okay. Now, if that concept is clear to all of you students, what if I tell you today I have animations for you? What if I tell you students that today I'll teach you fertilization with the help of animations? Sounds interesting? Sounds interesting, students? Today your entire fertilization is with the help of animations, which I have later on, which I'll show you also later on. Okay. So before we go to the entire fertilization, can we do something called as Microsporogenesis now? Sorry, Megasporogenesis. This micro and mega, I'm getting confused from that time. Can we start, students, now? Can we do Microsporogenesis now? Beautiful concept. Can we start? Yes, embryo sac is the female gametophyte. Like how pollen grain is the male gametophyte, embryo sac is the female gametophyte. In the case of anther, we have microsporangia. Microsporangium. Here the ovule, the entire ovule is Megasporangium. Ovule is the Megasporangium. Okay. Entire ovule is a Megasporangium. Now students, no. Let's start with something called as a Megasporogenesis now. Very detailed. I'll teach you. Do not get confused anywhere. If you get confused, ask me. Okay. Now, understand. What is Megasporogenesis? Process of formation of megaspore from the megaspore mother cell is called as Megasporogenesis. If you write this much, you will not understand anything. Okay. Let's go to the detailed part. Okay. Now, let me ask you a simple question. Microsporogenesis starts from where? Where does the microsporogenesis starts from? All of you are aware that microsporogenesis starts from nucellus. No, not nucellus, from the sporogenous tissue. From the sporogenous cells, it starts. Microsporogenesis starts from sporogenous cells. But megasporogenesis starts from nucellus. Nucellus. But the main difference is that one cell here, only one cell, right? Towards, let me write up only, a lot of space. One cell from the micropylar region. One cell from the micropylar region. Very important, student. This is very important. NCERT is given here. One cell from the micropylar region. Okay. One cell from the micropylar region undergoes differentiation and becomes large. One cell from the micropylar region. Meet once. If you go back here and check here, this, let's say this cell here. From the micropylar region, this is micropylar, right? This is micropylar region, this is chalazal region. From the micropylar region, one cell becomes differentiated, one cell. Because in the case of your microsporogenesis, many cells can become, but here only one cell. One cell from the micropylar region undergoes differentiation, it becomes larger, and that cell is called as what? MMC. That cell is called as your, one cell is called as your MMC, megaspore mother cell. Megaspore mother cell, students. Now this megaspore mother cell, what does it go? Mitosis or meiosis? This megaspore mother cell, does it undergo mitosis or meiosis? It undergoes meiosis, both 1 and 2. Okay, but it undergoes meiosis, students. It can give rise to 4 spores here. It can give rise to give rise to 4 spores here. The interesting thing to note here, students, that out of these 4 spores, only one is going to be functional and remaining 3 will degenerate. But the thing to know here is that, right, why this one will be functional? Why can't this be functional? Top 3 can degenerate, no? Or does it always happen like this? That is the main thing. Student, listen to me very carefully. Out of these 4 megaspores that formed here, the functional megaspore is always towards the chalazal side one. The chalazal side one is always functional. Remaining 3 with this one is towards micropylar one. Micropylar end. Right? All 3 will degenerate towards micropylar one. The one towards chalazal end. Let's say here we have the nucellus. Here one cell is undergoing. We got megaspore here. We got here. Okay. Now we have 4 megaspores here. Okay, 4 megaspores, right? The one towards the chalazal side is functional. The 4 towards the micropylar end, all of them will degenerate. That is the main concept here. How many of you understood? Tell me yes or no in the chat quickly. Quickly in the chat, tell me in the chat quickly. Quickly tell me in the chat. These are called as megaspores. And this is haploid in nature. So functional one is always towards chalaza. And here, students, do you notice here? Out of the 4, only one is developing. So can I call it as monospore development? I will call it as what? I will call it monosporic development. Monospore development. Okay. Students, do you think all of this is very difficult to understand? Tell me yes or no in the chat quickly. Very difficult to understand. Huh? So this is called as functional megaspore now. This is called as functional megaspore. Functional megaspore. Okay, functional megaspore we have now. Now what happens, understand, how does this functional megaspore develop now? How does it develop? Randomly, huh? No. Understand concept. Okay. Now here, let's say we have one functional megaspore. This is the functional megaspore. Now this functional megaspore will undergo first one mitosis. Okay, undergoes mitosis. Okay, now this mitosis, always remember students, it's supposed to be free nuclear. Why free nuclear, students? Because in free nuclear mitosis, what happens is there is no cytokinesis. Cytokinesis is not happening. There is no cross-wall formation. Only nucleus is dividing here, but there is no cytokinesis. Okay, that's called free nuclear. So this one cell now will become 2 cell. 2 cells. Now what if there was normal cell division, normal mitosis? If there was normal mitosis, you will end up getting 2 separate cells. But that is not happening here. It is free nuclear. Okay. Now students, which color? Okay. Now we have second mitosis. This is second mitosis. When there is second mitosis, these 2 cells will become 4 cells. 4 cells. Done. Now they have third mitosis, M3. Third mitosis. In the third mitosis, there will be 1, 2, 3, 4, 5, 6, 7, 8. There will be 8 cells here. 8 nucleated structure, 4 nucleated structure, and 2 nucleated structure. Done. Now, is this the end of it? Do we get end of it here only? No. This 8 nuclear structure undergoes polarization now. When it undergoes polarization means rearrangement. 3 will go on top, 2 in the middle, 3 will come at the bottom. That is called polarization. Now, once there is polarization, students, 3, 6, 7, 8, next step is cell wall formation. Now, what happens in cell wall formation? In cell wall formation, what happens is all the cell here will form a cell wall like this. All the cells here will also form a cell wall like this. The 3 cells here and the 3 cells here and 2 polar nuclei in the center. Okay. This will form one central cell like this, something like this. The 3 cells here, students, they are called as antipodal. And this is towards chalaza. chalazial side. And these two cells in the middle is called as polar nuclei. Polar nuclei, which will fuse together to form secondary nucleus. Secondary cell. Fuse together to form secondary cell. So two polar nuclei will fuse together to form the entire nuclei. Okay, and these three cells here, two are called as synergids. This is called as the egg apparatus or egg cell, and this egg cell and the synergids together they are called as egg apparatus. Egg apparatus. And that's how we get the entire female gametophyte, that is called as the embryo sac. This is the embryo sac. Here remember students, this is haploid. The central cell is diploid in nature. Okay, and students, can anyone tell me how many nuclei do I have here? How many nuclei? 1, 2, 3, 4, 5, 6, 7, 8. I have here totally 8 nucleated condition. How many cells? 1, 2, 3, 4, 5, 6, 7. This was one cell. I have 7 cells. So what do I get here? 8 nucleated, 7 cell structure called as your entire embryo sac. Students, do all of you understand? I'll give you a small recap. Yes or no? See, what happens in megasporogenesis? In the case of megasporogenesis, we have nucellus, which is diploid. This nucellus will give you the MMC. MMC is also diploid here. Megaspore mother cell undergoes meiosis 1 and 2 to give rise to your 4 megaspores here. 3 will degenerate. 1 is functional. Is towards the chalazial side, that functional megaspore, chalazial side. Nucellus, which nucellus is going? One cell from the micropellarent is becoming the MMC. Okay, this functional megaspore undergoes mitosis 1, mitosis 2, mitosis 3, give 4, 8 nucleate structure. Then it undergoes polarization. Here we have the 3 here, 3 top, 2 in the middle. Then cell wall formation happens. Cell wall formation is happening. Chalazial side, on the top. On the bottom we have the micropylar part side. How do you remember it, sir? AC. A antipodal, C chalazial. AC. Okay. Here we have micropylarent, we have synergids, 2 synergids and 1 egg cell. Together they are called as egg apparatus. Middle we have 2 polar nuclei which are joined together to form the central cell. Now students, in the synergids here, we have some cells here, tissue here, called as filiform apparatus. We have filiform apparatus here. You can see here. Can you see all of you? Nucellus is here. Nucellus. One of the nucellus towards the micropylar part is becoming the megaspore mother cell. 2-celled, 4-cell. 2-cell, 4-cell, 8-cell. Polarization is happening. Antipodals, 2 central cell. Then we end up getting the entire embryo sac. Towards the chalazial side we have antipodal cells. Towards the micropylar side we have the synergids and the egg. Together they are called as what? Egg apparatus. Then we have polar nuclei, 2 polar nuclei joined together to form the central cell or secondary cell. Okay. Then here we have something called as filiform apparatus. Function, I'll tell you later on, because their main function is during fertilization. That is filiform apparatus right here. Beautiful. Tell me, how many of you understood megasporogenesis? How many of you understood the megasporogenesis beautifully? Just fire emoji in the chat. I love teaching this student. This is so easy concept. See here. So beautiful, right? I want you to make all of these notes. Please tell me you're writing this down. Please tell me you're writing all this down, because it's very difficult. You end up becoming lazy later on to not write all this. Yes. Done, students? Done? Guiding the pollen tube, I'll tell you where the guiding the pollen tube comes. I'll show you the animations. Okay. Now we come to a concept called as pollination. Okay. Now students, if you understood the concepts till now, all of you, okay, if you understood the concepts till now, the chapter is almost over. Why do I say that? Because this chapter, the concept understanding, anther structure, gynoecium, the sporogenous tissue, the megaspore, microsporogenesis is a difficult part. Congratulations to all of you if you understood till now the difficult part, because rest of the part chapter is child's play. And all of you agree with it. All of you agree with it. Rest of the chapter is so easy. You will sleep also and learn. It's so easy. Okay, that's my guarantee. Okay. So remaining part is going to be so easy. Right. Let's finish it in one go. Shall we? All of you. I'll give you a break after outbreeding device, after fertilization. I'll give you a break after fertilization is over. Okay. So can we start pollination now? Can we start pollination? All of you in the chat. Can we start pollination now? Chapter is not vast. Chapter is very simple. Nothing is there. Okay. Okay. Sir, these cell divisions are plant grown or still developing? All of this is happening when the plant is grown, because flowering is the adult stage. Flowering is the adult stage. Flower does not happen in the young plants. When the plant becomes mature, then the flowering happens. Okay. Everything is easy. Exactly. Okay. Now what is pollination? Very simple concept. Transfer of pollen grains from anther to the stigma. That's all it's pollination. Transfer of pollen grains. Listen to me, all of you. From here, this is the anther. Transfer of pollen grains from the anther to the stigma is called as pollination. Done. Called as pollination. Okay. Now pollination can be two types: self-pollination, cross-pollination. Difference you understand. In self-pollination, what is happening? Transfer of pollen grains from anther to the stigma of genetically similar flower. It means that self-pollination can happen on the same plant. Same plant. Self-pollination is happening. Right. I'll give a simple example. Wedding is happening within the family. You say no, your mama's daughter getting married. So wedding within the family is called as your self-pollination. Okay. Cross-pollination. What is cross-pollination? You are getting married to an American girl. If you're a boy, you are getting married to an American girl. That is called as cross-pollination. Somewhere in America you're going. Imagine you're sending your friend is studying in America. He gets settled there. Cross-pollination. Okay, that is called as cross-pollination. Genetically different flower. Okay. Now here we have something called as autogamy and we have gynoecim. Difference you understand. In the case of autogamy, let's say here I have a flower. Let's say here I have a flower. Okay. Here I have anther. Okay. If the pollen grain is transferring, from here to here only, from the anther to stigma directly, okay, from here to here, that is called as autogamy. Same flower. What is gynoecim? You are marrying Indian only, but some other state. Let's take me only for example. If I get married to some girl from Jammu Kashmir or some girl from, let's say, Gujarat, why is it called gynoecim? Because plant is same. We are part of India only. We are part of India. Okay. So what is happening? This is a plant. Here and here, I have one flower. Okay. Here I have flower. Okay. Here I have, let's say, I have stigma and flower. Let's say here I have anther. If the pollen grain is transferring from here to here, here to here, okay, what does it say? Different flower, same plant. India only, no, not American. India only. Okay, so I'm getting married to someone in India only. Different state. One India, different state is there. That's all. That is called as gynoecim. Okay. Now here, students, in your autogamy, right, there are two main changes which you need to understand. Okay. For example, students, understand. In your entire autogamy, there is something called as there are two main things which you need to understand here. One is called as chasmogamous flower. And we have something called as cleistogamous flower. What is the difference? This flower, let's say, is very shy. Very shy. Okay. This is very open. These are the flower. Your chasmogamous flowers will always open flowers. This cleistogamous flowers are very shy. That is, do not open at all. They do not open at all. Okay. Here, if they are not opening, students, imagine there is a wedding in the house, but unfortunately, you cannot get married to other state people. You have to get married only Brahmin. Oh, full extreme casteism is there. You are not allowed to step out of the state only. Then do you have an option now, students? Tell me. Do you have an option now to get married to someone from other state now? Not possible. Within the state only now. Within the state. Let's say Karnataka only. Tamil Nadu only. Here, the flowers do not open at all. You're not allowed to go anywhere. So here only what happens? Only self-pollination. Only self-pollination happens here. Only self-pollination. There is no chance. There is no chance. Okay. Done? Done? All of you understood? Here there are three examples called as CVO. What is CVO? Comminalia bengalensis, viola and oxalis. CVO, that is going to be a Commelina, okay, and we have viola and we have oxalis, Done. Three examples here. Now let me tell you, students, these three examples, right? These three examples are also for chasmogamous. What do I mean by that? The meaning is very simple, students. A flower can have both cleisto and chasmo, both are possible. Why? Some people are very rebellious, they'll run away. They'll run away from the house. Okay. They get married somewhere else. So these flowers can have both cleistogamous and chasmogamous, both are possible here. Let me show you a diagram. Okay. See, this is chasmogamous on top, opening. Cleistogamous underground. Okay. Now see here. This is a chasmogamous flower of viola. See, viola has nice open flower is there. But at the same time, a viola will also have a cleistogamous flower. Viola. Same goes for your Comminalia also. It can have an open flower, but it can also have a closed flower, underground flower. So here only self-pollination, here cross-pollination can occur. Tell me yes or no. Beautiful, no? Beautiful, right? Tell me all of you are understanding or not? Understanding? Cleistogamous, close. Chasmogamous, very open. Okay. Now students, what is xenogamy? What is xenogamy, students? What is xenogamy? Tell me. Xenogamy is you go to America, you never come back. Okay. My friends are there. Okay. They don't want to come back only now. Okay. So there is something called as xenogamy. In xenogamy, there is variations now. There is variations. Till now, in geitonogamy and autogamy, only Indian looking kids. Okay, kids are very Indian looking here. There is no mixing up genetic material is same because same plant. But as soon as we come to xenogamy, what happens? In the case of xenogamy, let's say I have one plant here, I have one plant here, I have one flower here, I have one flower here. Let's say here I have your entire. So if the pollen grains are transferring from here to different flower, that is called as different flower, different plant. Here we have different flower, different plant. In we have light skin kids, different plant. Okay, so different plant only here. I hope all of you are understanding. That is your xenogamy. Different countries here. Okay, xenogamy. Now students, this pollination, it doesn't happen its own. Not possible. There are something called as agents of pollination. When the pollination has to happen, there has to be some brokers. You know, right? In India also, there is a matrimonies. I'm shocked, students. There are something called as. First, I thought there is only Indian matrimony. No. Now there is matrimony for state also. There is matrimony for every language also. Matrimony for caste also. I'm like, where is this world heading? Okay. This is your matrimony. Okay. Agents of pollination. Agents of pollination or the broker. Okay. Now, pollination, it has to happen. It doesn't happen on its own. It needs a mediator. This is the mediator. Now, there are two types of mediators, students. There are two types of mediators. There is something called as biotic and abiotic. Biotic and abiotic. Okay. Biotic, we have your animals and insects. In abiotic, we mainly have wind and we have the water. Wind and water. Abiotic. Okay. So, clear, students? Clear? We'll study this now. Very simple. Now, I'll tell you what you need to remember here is, all you need to do remember here is the properties of wind, water and insect pollinated flowers. What are the properties shown by these flowers? Like every matrimony has its own this thing, right? Features. Let's say Bharat matrimony, you can upload photo, only girl can message first, and you have to buy premium version to do something else, right? To do some upload a video of yourself. So every matrimony has its own properties, right? Similarly, every single broker here, every single agent, biotic or abiotic, a flower has certain properties or features that you need to remember, and if you remember that, it's over. It's over. Okay. Now, can we start? Can we start? All of you? Yes. Now, what about agents of pollen? We have wind, water. Wind is called as anemophily. Water is called as hydrophily. Okay. Bird is called as ornithophily. Insect is called as entomophily. This one is cryptophily. That is for bats also is different. Bats, it is not given in your syllabus, so I didn't mention it. So, birds, insects, wind, water. Now, students, this is the entire image given here. This is for your wind. Okay. Pollination by wind is more common among abiotic pollinators. Wind pollination also requires that the pollen grains are light and non-sticky. First point. Okay. They even transport very wind currently. They possess often possess well exposed stamen. So stamen has to be exposed. Stamen is exposed. Pollination can happen very easily. Pollen grains are very light and non-sticky. Okay. Large, often feathery stigma. Stigma is very feathery to trap the pollen grains. Okay. A single ovule in each ovary, single ovule, not many, single ovule is there. Okay. Now, numerous flowers packed to form inflorescence. Many flowers are packed together from inflorescence. Stigma and style, which wave in the wind to trap the pollen grains, and it is common in the case of grasses and asteraceae members. Here is the properties. You need to remember only this. Small and inconspicuous flower can be there. If they're very small, they can be joined together. Colorless, odorless, nectarless flowers, right? Small inconspicuous flowers because it's wind pollinated. Flowers are packed in inflorescence, yes, because to go easily. Pollen grains are light and dry, non-sticky, non-dusky, well exposed stamen, feathery stigma, uni-ovulated pistil means single ovule in ovary. Single ovule in ovary. Any doubt here? Quickly ask me, students. Easy or not? Tell me, easy or not? Easy or not? Tell me, students. That's all you need to remember. That's all you need to remember. If you remember this for wind, your life is easy. Okay. Now, what about water? Pollination by water is quite rare in flowering plants and limited to 30 genera. So it is very less in the case of flowering plants, very difficult, they don't do it much. But who does it? Water pollination is done by your bryophytes, pteridophytes and algae. they do more water pollination compared to angiosperms. Okay, it's given here. It's given here. Now, what is the example here? Understand the example. Need for water for the transport of male gametes and the fertilization. Some examples of water pollination plants are Vallisneria and Hydrilla. I'll tell you this one. Which in fresh water. Vallisneria, Hydrilla, fresh water. Marine, we have Zostera for marine. Now, students, here I'll tell you one beautiful concept. Understand. Okay. See here. This is happening in the case of Vallisneria. What happens in Vallisneria? Can you see this is the female flower right here? Let's say this is a female flower, high and mighty here. Female flower is on top here. Now here we have male flower. Male flower is here. Now what happens in the case of Vallisneria? In the case of Vallisneria, what happens is male flowers are released, and these flowers, when they release the pollen grains, right, the entire pollen grains will go on top. Can you see here? They float on top. They'll float on top and they eventually go, and then they reach here. Pollen grains will reach here, and then pollen grains will dust on this, and entire pollination happens. The entire female plant, right, it will have a long stalk, and the flower will be on the top here. But male flowers are produced underwater, and pollen grains are released and they go on top. This is seen in the case of Vallisneria. Okay. In the case of Zostera, it is completely underground. Male and female flower both are underground only. In case of Zostera, can you see here? Long stalk, flowers are going on top here, on the top. Can you see female flower is present on the top here? Long stalk. Okay, long stalk. See, this is Vallisneria. This is Hydrilla. Even Zostera also like this. Zostera also is completely underground. It is completely underground. Okay. Okay. Now students, there are always some mental people, no? There are always some people who they're very against everything in the world. For example, they can have all the resources in the world and then still not study. Yes or no? There are people who will have all the resources in the world. They will have all the classes with them, all the notes with them, all the mock tests with them. They will still not study. Okay. I'll tell you one more example is given on the board right now. There are certain plants which are present in water. But they do not do water pollination. They do not do pollination with the help of water. There are two mentals like this on the board now. Mentals. I'll show you. Not all aquatic plants use water for pollination. There is something called as water hyacinth and water lily. These two plants are growing in water, but they don't do water pollination. Simple, no? They just use the water. They do not, like, no, I will not use water. I will use insect. I will use wind. Extra work they want to do. Extra work in life. Okay. So the flowers emerge above the level of water and are pollinated by insect or wind. Tell me mental or not now. Tell me. Tell me now, students. Tell me. Okay. Now, in Vallisneria, the female flower reaches the surface of the water by long stalk, and the male flower or the pollen grains are released onto the surface of water. Okay. See, in seagrass like Zostera, female flowers remain submerged in water and pollen grains released inside the water. Now here, students, a pollen grain will have something called as mucilage covering. Why? Mucilage covering is basically waterproof paint. You know what is waterproof paint? It is like a coating which they do, proper coating, right? Water coating they'll do, and pollen grains will not become wet. Okay. No. This is your mental here. All of you see, look at it, look at that person, look at this person. Look at this person right here. Okay. Present in water everywhere. Okay, but still doesn't want water. Insect pollinator, water hyacinth. Okay. Majority of the flowering plants use a range of animals. Students, here, this is very important line I told you. Important line here is mucilage covering. Apart from that, one more important line here is, which I missed out by unfortunate means, right? That is, pollen grain in many species are long and ribbon-like here. Okay. Long and ribbon-like pollen grains. This also is a very crucial point here. Long ribbon-like, mucilage covering. Two main points here. Okay. In majority of the flowering plants, they use bees, butterflies, wasp, beetle, ant, wasp, everything is there. Okay. Bees are dominant biotic component pollinating agent. Compared to all of this, bees are the most important ones which help in pollination. Bees. All of you know bees. There's a saying that if all the bees disappear from earth, the entire earth will become extinct in the next decade very easily. Okay. No. There are other animals also, lizard, gecko, everything is there. Okay. Now students, let's come to flowering plants now. Okay, these are show off people. All of you know there are show off people in the world. Who will show off? I know this, I know that. There are show off people. I have new jacket, I have new car, I have new bike. These are called as show off plants because they will do whatever possible in the world. They will do every day new jacket, every day new shoe, show off this new car, new bike every day. Show off people are here. So flowering, the flowers, the plants which produce bright flowers are show off because they are showing off for whom? For the pollinators to come. Okay. So let's look at the show off people. Okay. Let me drink some water, students. Throat is giving up little. Now, who are the show off here, students? Show off. Color. All the insect pollinated flowers are show off. Large, colorful, fragrant and rich nectar. New jacket, new perfume. On Zara perfume they'll go buy. Puss everywhere. Okay. Whole world will get to know they have put perfume today. Okay. Full headache will come. Okay. So nice, and they have fragrant and rich nectar. Okay. Nice nectar. They bring very good tasty food. Unfortunately, even I have fallen into this trap. There used to be a friend who used to bring tiffin, used to be so nice. I became friends with him. To eat his tiffin. I became trapped here. When the flowers are small, a number of flowers are clustered in the inflorescence. If many flowers are not seen very well, what do? They form a group now. You have seen show off people always travel in group. You might have noticed in school or college. All these show off people have their own group. Yes or no? Tell me yes or no. You tell me in the chat now. All these show off people are always found in group because they become more bigger. They shine brighter. Okay, they shine brighter. Okay. Animals are attracted. See, show off is working now. Attracted to the flower by color or fragrance. Poor people. The flower pollinated by these flies and beetles secrete foul odor. See, there are two extremes. One nice smell. There's one more group of people. I will not take bath in 10,000 years. All my life I will not take bath. Okay. Now all of these people who do not take bath will all group together. Okay. Now there are some flowers which will give a rotting smell. With the help of this rotting smell, what happens is all the flies will come and go there and pollination happens. So they have different target audience, different target. Flower which has very bright smell, honeybee. Bad smell flower, flies. So they know their audience. Okay. Now, nectar and pollen grains are usually floral reward. They give reward also. If a pollination is happening, they'll give nectar, they'll give pollen grain to reward. Reward mechanism is there. The poor honeybee, bumblebee, thinking that something good is happening in life, it'll keep on coming there now. It has fallen into a trap now. For harvesting the reward from the flower, the animal visits come in contact with the anther and the stigma. The body of animal gets coated with the pollen grains. Now students, you tell me in the chat. If pollen grains has to be coated on the animal, the pollen grain has to be sticky or non-sticky? It has to be sticky. Then only get attached there. So sticky pollen grains, right? Which are generally sticky in animal pollinated flowers. Okay. When the animal carrying the pollen on its body comes in contact with the stigma, it brings about pollination. Here are the properties which you need to remember. That's all. Showy and bright color, show off. Small group together, conspicuous. Landing platform or honey nectar guiding. Odor or foul odor. Secretion of nectar. Edible pollens. Reward mechanism. Students, tell me, are you understanding or not? Students? Tell me yes or no. Okay. Yeah, obviously. Now students, now there are this people in this world who are extra smart. Okay. They will go here, they'll go there.
They will take the food, they'll run away from there. They are called as nectar robbers. This is the kalla here. This is the kalla here. Okay. This is called as nectar robber. What will this person do? This person, let's call him, Sam. Okay. His name is Sam. Okay. This Sam here is very notorious. Why you say? He will go and take the nectar, but never pollinate only. Okay. So it is taking all the goodies in the world, but not doing any work. Okay. Don't be Sam. Be some useful in life. Like the video. Okay. Now students. Now, how do we prevent people like Sam, students? How can we prevent? How can we prevent people like Sam? Like, some flowers, they'll be like, I know you are dangerous person. I know you are Sam. So what do? Now they will do something called as secret code. They will do something called as secret code now. This code will be only available to a certain honeybee and certain flower. They'll exchange the code secretly and then only they will benefit. And that code will not be told to Sam. They will like, please do not tell the code to Sam. They'll tell the code, please do not tell the code to Sam. And that is happening. That is happening in the case of your animal kingdom. That is called as mutualism. Right? Some species floral reward are in providing safe space to lay eggs, that is there. Okay, for example, tallest flower, Amorphophallus, the flower itself is about 6 feet in height. Right? Here, this is Amorphophallus, 6 feet height. Okay, Amorphophallus here. Okay, they'll provide laying egg space. A similar relationship exists between a species of a moth. Okay, there is a special moth called as Pronuba moth. Okay, moth and yucca. Both of them have a special code. They have a code here. And this code they will know. So both of them have a mutualistic relationship. This moth can come here and lay the eggs and also help pollination. No other intruder can come which does not have a code. Understood all of you? Where both species, moth and the plant, cannot complete their life cycle without each other because only both of them know the code and they will complete the life cycle with each other only. Okay. The moth deposits egg in the locule of the ovary and the flower in turn gets pollinated by the moth. The larvae of the moth come of the egg and the seed starts to develop. So similar thing which you have learned it in your ecology also. Wasp, fig and wasp. Fig and wasp also you might have learned. Fig and wasp. But they share a secret code, only they can go over them. So with the help of this method, they can avoid any type of intruders or pollen robbers or nectar robbers. Students, how many of you understood the concept? How many of you understood the concept? Tell me in the chat quickly, all of you. Understanding or not? All of you understanding? Do not doubt. All of you understanding the entire pollination. So here you need to remember, the only thing you need to remember here is the entire properties of different types of pollinations and you're good to go. Okay. Now a question in front of all of you. A question on the board. What happens when self-pollination continues throughout many generations? Imagine there is a very bad character present, undesirable character present in a plant. That undesirable character will keep on passing every single generation. That eventually leads to the decrease in the fitness of a flower. The sexual reproduction capacity decreases. That is called as inbreeding depression. It causes inbreeding depression, students. It causes inbreeding depression. Now, if you do not want inbreeding, you're like, stop inbreeding. How can we stop inbreeding depression, students? Is there a way to stop inbreeding depression? Yes, there is. Yes, there is. The inbreeding depression can be stopped by something called as outbreeding devices. Now, what is outbreeding devices? Answer is very simple, students. For example, let's take I have a flower here. Flower here. Now this is a bisexual flower. Bisexual flower is there. Okay. Yes, they also have depression because they're seeing the same face every single day. Okay. Now here we have the bisexual flower. Now what if this bisexual flower is not anymore and this becomes unisexual? So a plant where there is a plant, entire plant only has a gynoecium, will have a female plant, and we have separate male plant. Female plant, male plant. Then depression is gone. That is called as unisexuality or unisexual flowers. Unisexual flowers. We can have female plant and male plant separately. Next is called as dichogamy, students. Dichogamy. What is the meaning of dichogamy? Imagine. There are two people. Okay. In that, we are rejecting the marriage here. Why? The girl is ready now, but the boy is not ready. Like very immature. Brain is not developed only. Okay. What if the boy is ready now? Yes, I want to get married, I'm responsible. But the girl is immature, the girl is not ready. Okay. So in such cases, the marriage is not a possibility. Same concept also applies in dichogamy. In the case of dichogamy, what happens? Sometimes stamen is mature, stamen is ready, but the gynoecium or the pistil is not ready. Or pistil is ready here, but stamen is not ready. All of you understood? All of you understood what is dichogamy? Tell me yes or no, quickly all of you. Stamen can be ready, pistil cannot be ready. That is, it's immature. Or pistil is mature now, or stamen is not mature anymore. That's all is called as dichogamy. Now what is self-incompatibility? Self-incompatibility is the condition where the plant is like, I am done with life. I do not want self-pollination. It will change its genetic composition only. The entire genetic is changed. It's a genetic condition. Where it becomes self-incompatible. Like, I am done for life. I do not want self-pollination anymore. It will change the genome itself. Okay. That is self-incompatibility. Okay. What is heterostyly? Understand. Here, simple example I'll give you. The girl is very tall. Let's say the girl is around 6 feet 6. Girl is 7 feet tall girl. Okay. She is now getting ready married to, let's say the boy is, is 4 feet tall. Boy is 4 feet tall. Girl is 7 feet. Boy is 4 feet. They'll say, excuse me person, marriage is not possible because I am very tall, you are very short. Marriage is rejected. Or the boy is, let's say, is 7 feet, the girl is 4 feet. Even though it happens, marriage happens, but here we'll say not accepted at all. Marriage is again rejected because of height. So marriage is rejecting because of height now. Okay, same concept to apply here. What happens in heterostyly here is, let's say this is your entire gynoecium and let's say style is here. The style is right here. The style is the entire stamen is here. Pollination cannot happen easily. Rejected. Okay. One condition. Second condition I'll tell you. Here we have the gynoecium. Okay. Second condition. Let's say here I have the gynoecium and here I have the anther like this. Here we have anther. Is it possible? No. This is called as heterostyly. Okay, heterostyly. So because of heterostyly, self-pollination can be prevented. It can be completely stopped and we will end up getting cross-pollination happens, and these are called as outbreeding devices. Unisexual flowers, dichogamy, self-incompatibility, heterostyly. Different in style. Dichogamy is different maturity time and self-incompatibility is genetic factor. Okay, tell me all of you understood or not? All of you understood? Outbreeding devices? Understood or not? Really, students, understood? Understood or not? Students, examination, please write this. Don't write my story. Okay? Don't write, so heterosexually girl is very tall, boy is very short. No, don't write that. Do not write this in your board exam also. If you're preparing for board examination, please do not write this in examination. They will think, where did you even learn all this from? Learn, write this, draw this and tell them in board exam at least. Okay? I don't want to get in trouble. Okay. Next is called as artificial hybridization. What is this concept? Very simple concept. Okay. Students, how many of you love mangoes? How many of you love mangoes? How many of you love mangoes? You love mango? I love mango. Okay. Let's take two mangoes here. Let's take, I have one mango 1. Mango 1 and I have mango 2. Mango 2. This mango 1, very small mango, but very sweet mango, short and sweet. Okay, very sweet mango. This is very large mango, but not sweet at all. Not sweet. Okay. Okay. No. Mango 1 small, very sweet, very large, not sweet. Okay. So what do? If you do self-pollination here, same traits will be carry forward. What do we do now? We take mango 1, we take the plant here, we take this plant here. Let's take, we stick the pollen grain from here, and from here we take the entire pistil. Okay, where we can find the entire stigma cell ovary. We take this entire pollen grains and we send it here. We do this manually. And this is called as artificial hybridization. Why? Because the offspring now will be very sweet and very large. This possibility is there now. Very sweet, very large. But is there a possible that we end up getting very small mango and not sweet at all? Not sweet at all? Yes. This is also possibility. This is also possibility is there. Small and not sweet. This possibility is also there. This possibility is also there. Okay. So you have to be very, luck should be there. Luck is there, will happen. This is called as artificial hybridization. Now here the techniques are here. Okay. Now how do we do it? There is something called as emasculation, bagging, rebagging, and there is something called as tagging also. Tagging. Now what is emasculation? For example, if I have a bisexual flower, in the case of remasculation, we have to make sure we have to remove this anther here. Remove all the anthers. Why? If you don't remove the anther, pollen grain can go here, So no use. So what do? Removal of anther before dehiscence. We completely remove all the anther to make sure self-pollination is not happening. But do we do this in unisexual flowers? No. We do not do this in unisexual flowers because they are already unisexual. Why? To make them more unisexual. Okay. So we do emasculation in bisexual flowers. Next we do something called as bagging. Why do we do bagging? We do bagging because we do not want any other unwanted pollen grain to come and land here. So we do bagging. Okay, bagging. Now what do? Next, we remove the bag and the whichever pollen grain which we need, we add the pollen grain and we do rebagging. Why do we do rebagging? We do rebagging because to make sure the entire process happens and there is no more stoppage there. No more unwanted pollen grain is coming. Then we also do tagging. What is tagging? In tagging, we put a name card there. We write the plant name, which pollen grain from where, which stigma from where, right? When was it pollinated? All the details is given there for tagging. That's all. Name tag is given. That is called as your artificial hybridization. Now students, here's a question to all of you. Can the artificial hybridization between any species Can I take a monkey and a donkey? Can I take a monkey and donkey and make some the monkey? Is it possible? Can I do the monkey? Not possible. Can I take a tomato and a potato and make pomato? Somatic hybridization, yes. Somatic hybridization we can. But in the case of normal hybridization, we cannot. It is not possible at all. Why? What is the main reason why we cannot take a tomato and a mango and make a mangapto, something like that? Not possible at all. Not possible at all. Okay. The reason is that because every single time a pollen grain lands in the stigma, it has to write a test there. It has to write a test. Okay. Test. What is the test here? The test here is called as pollen pistil interaction. For example, this pollen grain is coming here. Pollen grain is coming here. On the stigma. Now it has to exchange some dialogue. There's a dialogue here. Pollen grain will say, please let inside, please let me go inside. This pollen grain also will say, please let me go inside. But this pollen grain is very good. We are like, oh, same species, oh, my relative only come inside the house. But this one is a kalla. This one is a, let's say, is a thief. Will let a thief come inside? No. Rejection will happen, rejection will happen. Relative come inside the house. Okay. So what is happening here is, there is something called as, once the pollen grain lands on the stigma, there is a chemical dialogue exchange. If the stigma and the pollen grain are from the same species, then acceptance is happening. If they are not same species, rejection is happening. So if the acceptance is happening, pollen tube formation starts. If there is a rejection, no pollen tube formation. That is the starting of pollen pistil interaction. Then what happens? Once the pollen grain lands here, it will absorb all the moisture and it will swell up. After swelling up, students, the intine is coming out. Remember, students, I told you in starting, this is your entire pollen grain. Here we have 𝐂𝐡𝐞𝐤. Here we have the entire 𝐂𝐡𝐞 here. Now what is happening here? Through this small spot called as germ pore. So small spot called as germ pore, the entire intine is coming out in the form of a pollen tube. Entire intine is being pushed outside. As a pollen tube. So pollen tube formation is happening. Okay. Yes, relative also we say don't come inside, but okay, how can we say? Students, don't tell that in the house. You tell when the relatives have gone. Do not tell when the relatives are coming here, don't come inside. Don't tell that. Okay. If you tell that, I know, inside you want to tell. Inside your mother also wants to tell the same thing. But you cannot tell it openly. Okay. Now, there are multiple steps here. Okay. The contents of the pollen grain move into the pollen tube. So all the contents, what do we have? We have the generative cell, we have the tube nucleus, that is the vegetative cell. Can you see? Vegetative cell is in the front here. Can you see? Vegetative cell is in the front here. It will completely guide it. And here, generative nuclei, that is, the generative cell will divide into two cells now. These are called as two male gametes. Two male gametes are produced here. These are two male gametes are released here. And they are going, two male gametes. Okay. So pollen tube formation is happening. Can you see pollen tube formation is happening and it is entering here. It is entering now. Now students, listen to me very carefully, all of you. Listen to me very, very carefully, all of you now. Students, can you see? This is your three antipodal cells. These three are your egg nuclei, two polar nuclei. Okay. Now students, understand what is happening here. These are your, can you see, synergids and egg cell. Okay. This is your anther. Here we have pollen grain. Pollen grain is released from here. Okay, it has something called as this anther. This pollen grain has something called as your generative cell, vegetative cell. When vegetative, two generative nuclei now. Okay, now pollen grain will go. It will land on the stigma now. Can you see it's going? Land on the stigma. Once it lands on stigma, what happens? Once it lands on stigma, what happens is can you see the vegetative cell is going front? Can all of you see? This entire thing is a vegetative cell. In the behind, we have the two polar nuclei. This right here is a pollen tube formation. This is the entire vegetative nuclei, and there are two generative on the back. Okay. Can you see? Just see this. That is entering through style. It is entering inside now. Okay. Now this is the entire thing is called as your embryo sac. Okay. Now listen to me very carefully, all of you here. Entry of pollen tube inside ovule. First concept. Entry of pollen tube inside ovule can happen from three different places. Inside ovule. So entry of pollen tube inside ovule can happen from the micropylar end, from the chalazal end, or from the integuments. Entry of pollen tube inside ovule, three places: chalazal, micropylar, integument side. But entry of pollen tube inside embryo sac, always micropylar end. I'll repeat again. Listen to me very carefully, all of you. Listen to me very carefully. Entry of pollen tube inside ovule. First of all, ovule. Female gametophyte, the Megaspororangium. It can happen from the chalazal side, micropylar side, or integuments. But entry of pollen tube inside embryo sac, the female gametophyte, it is always through micropylar. Why? Because near the micropylar we have the egg. Okay. Now see here. It is entering. Now let's say it is entering through micropylar now. Can you see it is entering here? Pollen tube is entering. Now once the pollen tube enters here, inside where? It is entering through the synergids. Okay. So the pollen tube is entering. It is entering through micropylar end. And it is entering through the synergids, and once it's enters inside the synergids, the pollen tube will burst open. Okay. Now before we go to the next concept, let me tell you here. See, can all of you see this? Entry into embryo sac. This is entry into embryo sac. Always through micropylar. Why? Because egg is present near micropylar. Can you see here? The pollen tube is entering. Vegetative nucleus is disintegrating now. No more use. Male gametes are released inside. This one is called as degenerating synergid. With the help of filiform apparatus, entering inside. Okay. Now, starting from pollen tube formation or stigma landing, pollen grain landing on stigma, from there to here, this entire sequence is called as pollen pistil interaction. Entire thing. All the way from the pollen grain landing on stigma until here, everything is called as pollen pistil interaction. And I told you here, there are three main types. Let's see, there is something called as porogamy. What is porogamy? Porogamy is pollen tube entry inside through micropylar side. And all of this is for ovule. Remember that. Entry of pollen tube inside ovule. First of all, ovule can be of three types: porogamy, chalazogamy, mesogamy. It can enter from here. Can you see it? Enter from micropylar part. Done. Pollen tube can enter from the chalazal part also. It can also enter from the integuments, from sideways. That is called mesogamy, chalazogamy, chalazal side, and porogamy through micropylar. Now this is entry into the ovule, but entry into the embryo sac. Entry into embryo sac is always towards the micropylar. Okay, towards the micropylar only. Okay, so pollen tube is entering here. See, entry into embryo sac is always through the micropallar region into embryo sac. Why? Because the egg cell is present here. Egg cell is present here. That's why it's entering here. Okay. So this entire thing is called as pollen pistil interaction. All the way from the pollen grain landing on stigma till here. Entire thing is pollen pistil interaction. All of you understood? All of you understood, students? Clear, students? That is called as pollen pistil interaction. Now, what is exactly happening? What is exactly happening once the pollen grain enters inside? Once the pollen grain enters inside, what is happening? For example, students, here is the embryo sac. Let's take embryo sac. Let's take embryo sac here. Here towards the chalazal side we have 3 antipodals. Here we have the 2 synergids and 1 egg cell. This is the egg. This is 2 synergids. Yeah, 2 synergids. 2 synergids are here. Now what happens once the pollen tube enters inside? What happens? What happens when the pollen tube enters inside? It is always entering from here. Pollen tube is entering from here. Okay. Now here we have the filiform apparatus. Filiform apparatus. Now once the pollen tube enters inside, it will burst open and it will release 2 male gametes. One gamete and second gamete. Okay. Now here we have the central cell. Now this first male gamete. Okay. The first male gamete. It will fuse with the egg and that will result in the zygote formation. Male gamete is haploid, egg is haploid, zygote is diploid. This entire thing is called as syngamy. Now, what about the second male gamete? Second male gamete will move up a little. Second male gamete. The second male gamete will fuse with the 2 polar nuclei, also called as central cell, and this will result in formation of PEN. Now this second male gamete is haploid. This is diploid. So PN is going to be triploid. N2N plus N is 3N. This PN eventually becomes primary endosperm nucleus. Primary endosperm nucleus will become PEC, primary endosperm cell. And this PEC will eventually develop into endosperm. And this entire thing is called as triple fusion. Triple fusion. Why triple fusion? Because 3 nuclei are joining here. 3 nuclei is joining here. Now students, here we have, it's called as double fertilization. It's called as double fertilization because there is syngamy happening here and also there is triple fusion is happening here. There are 2 main things are happening here, right? Triple fusion as well as syngamy. 2 types of fertilization is happening here. That is why it's called as double fertilization. That's why it's called as double fertilization. That's why it's called as double fertilization. Is that point clear to all of you, students? Is this point clear to all of you? First male gamete, egg, zygote. Second male gamete, polar nucleus, central cell, and PN, PEC, endosperm. Triple fusion, syngamy. Triple fusion and syngamy, and that is why it's called as double fertilization. Clear, all of you? Clear? Clear, students? Triple fusion, syngamy. Triple fusion leads to endosperm formation, zygote formation. Students, with that, we come towards the end of the chapter. With that, we come to the end of today's chapter, and we are only left with something called as post-fertilization events. That's all. Here we will notice that how zygote will develop into embryo. Here we'll notice that how the PN will develop into endosperm. We'll notice that how ovule will develop into seed, and finally how ovary will develop into fruit. This is the last step, right? This is the last step in today's class, post-fertilization events. Nice. Friends, nice or not? Tell me. Now students, we will take a 5 minutes break and we'll start, shall we? Let's take a 5 minutes break now. Yes. Cash is not in cash counter, cash is simply sitting somewhere. Okay. Now what's the time now? What's the time? Time is 11:53. Let's just 7 minutes break. Class will resume at 1 p.m. Okay. So we will take 7 minutes break, hardly 7 minutes break. So I will go fill some water and come. My throat also let me rest for like 7 tala for reason exactly again. What? Okay. So let's take 7 minutes break. I'll be back in 7 minutes. Okay. Sayonara. Sound check. Yes. Welcome back, students. Welcome back. Welcome back. Now, we come towards the end of the chapter. That is, last thing is called as post-fertilization events. The concepts here are actually very, very easy to understand. Okay, let me tell you. Okay, one by one. Now, first of all, endosperm. Okay, all of you know endosperm is formed as a result of triple fusion. Triple fusion. But what is the main function of endosperm? Can anyone tell me what is the main function of entire endosperm? Right. The main function of endosperm is nothing but it provides nutrition to the growing embryo or the growing cotyledon. It provides nutrition. Okay. Now there are two types of endosperm. One is cellular, one is cellular, one is free nuclear. What is the difference? Let me teach you. Okay. See, these are the two different types of endosperm. One is called as cellular endosperm. One is called as free nuclear. Free nuclear endosperm. Understand the difference, students. In the case of free nuclear, do you see any type of cross-wall formation? Do you see? Not at all. So in the case of free nuclear endosperm, there is no cytokinesis. In the case of free nuclear, there is increase in nucleus. Can you see increase in nucleus? That's why it's called as free nuclear endosperm. But at the same time, if you look at the cellular endosperm, in the case of cellular endosperm, we can always see cross-wall formation. Cross-wall formation. That is the difference between free nuclear and cellular endosperm. Okay. Now let's see what is given here. Okay. Now if you look at a coconut, right? Here in the coconut water which you get, the coconut water is free nuclear endosperm. Right? Coconut is inner multinucleated free nuclear condition. Here we have multinuclear condition. Why? Because here one cell will have multiple nucleus. Because there is no cross-wall formation, because of no cross-wall formation, multi-nuclear condition will arise here. Okay. Now what about white kernel, this white part? The white part, outer multicellular solid endosperm. This white part is basically cellular endosperm. Cellular endosperm is given here also. See, PN undergoes successive nuclear divisions to give rise to free nuclei. This stage endosperm develops is called as free nuclear endosperm. So there is successive nuclear division is happening. There is no cross-wall formation here. See, subsequently cell wall formation occurs and endosperm becomes cellular. So it is becoming cellular only after cross-wall formation. Only after cross-wall formation. Okay. The coconut water from the tender coconut that you are familiar with is nothing but free nuclear endosperm. The water is free nuclear endosperm made up of thousands of nuclei. Can you see here? It is made up of thousands of nuclei here. Thousands of nuclei like this. Thousands of nuclei. Thousands of nuclei. Okay. Thousands of nuclei. The white kernel is cellular endosperm. Cellular endosperm. Because in the case of cellular endosperm, there is cross-wall formation is happening there. Cross formation is happening. Now, what if endosperm is completely consumed before seed maturation? For example, seed is maturing. If the endosperm is completely consumed, it is called as non-albuminous seed or non-endospermic seed. It is called as non-endospermic seed. Okay. Where is it given? Where is it given? I wrote it somewhere. No. Sure. Albuminous seed means what? Endosperm is present. That is called albuminous. What is ex-albuminous or non-endospermic? Endosperm is absent. The endosperm is completely absent. Example is pea and groundnut here. Pea and groundnut here. Okay. Albuminous is wheat, maize, cereals. Basically, all of them are monocots mainly. Are here albuminous seeds. Albuminous. It means endosperm is present here. Endosperm is absent here. Endosperm is absent here. Okay. Endosperm is absent. Now, understand what is the meaning of embryo and how does the development of embryo happens. Now, once the zygote is there, right? The zygote will start to multiply now. Yes or no? Zygote will start to multiply. When the zygote starts to multiply, that gives rise to the entire embryo eventually. See the diagram, you understand better. Okay. For example, this is your unfertilized egg. Okay, fertilized egg. This one is your egg right now. This is the zygote here. Zygote. The zygote, once it starts to divide mitotically, free nuclear division. Okay, it will give rise to two cells now. Two cells. Now here, when this give rise to two cells, one is called as apical cell on top, bottom is called as basal cell. This apical cell is also called as terminal cell in many cases. Okay. Now students, focus. Now this apical cell right here, and we have basal cell right here. Now can you see, this one is forming the above head, the bottom one is dividing and giving the entire, one second. Here basal cell will remain as such. Can you see, this is a basal cell. This apical cell will start to divide. This will form a long suspensor-like structure. Okay. Here we have the basal cell. Here we have the entire apical cell. It will keep on dividing. So it becomes octate, globular, triangular, heart shape. This is the entire thing suspensor. Then torpedo and mature embryo. In your NCERT, you have only learned with, I think you only have globular, heart shape and mature embryo. This is also called as, octate is also called as proembryo. Proembryo. Embryo before formation. This is the entire development of the embryo. Development of the entire embryo. Okay. You know, I don't think you have torpedo. Torpedo is not given you a syllabus, right? So you have mature embryo directly, which has two cotyledons. Okay. Let's see here. See, embryo develops at the micropylar end of the embryo sac. Why? Why, students? Because at the micropylar side we have the egg. Zygote formation is also happening in the micropylar part. So if zygote is near micropylar, embryo will also form at micropylar only, obviously, right? Most zygote divide only after a certain amount of endosperm is found. Very important line. Why? Because zygote will always wait. Zygote doesn't change to embryo because it needs nutrition. The division needs nutrition. So until endosperm is formed, which can technically provide nutrition, zygote will not divide. So endosperm formation happens first, then embryo formation. Okay. This is an adaptation to ensure assured nutrition to the developing embryo. Through the seed differ greatly. Early stages of embryo development, embryology are similar in both monocot and dicot. It is same to same almost. Okay. Now, the zygote give rise to the proembryo. I told you octate structure, proembryo, and subsequently give the globular, heart shape and mature embryo. Globular, heart shape and mature embryo eventually which you get. Okay. Now, a typical dicot embryo consists of an embryonal axis. For example, students, see this. This is your two cotyledons. Here we have the entire embryonal axis like this, embryonal axis. In this entire embryonal axis, students, let's say this can give rise to shoot and this can give rise to root. The just below the shoot, this region is called as epicotyle. Just above the root, this region is called as hypocotyle. Epicotyle and hypocotyle. Okay. So embryonal axis and the two cotyledons. These are the cotyledons. These are the cotyledons right here. The portion of embryonal axis above the level of cotyledon is called as epicotyle. Above is called as epicotyle. Below is called as hypocotyle. Okay. If there is root here we can find root cap also. We can find root cap here. Root can be found here at the bottom of the root. Okay. The root. Now, if you can see, this is your entire embryo development here. Plumule, we can see hypocotyle here. Above radical, we can see hypocotyle here. If plumule is here, we can see the epicotyle there. Epicotyle here. Okay. This is also the embryo here. We can see here we have scutellum here. So if the scutellum is present here, students, can anyone tell me, is this going to be dicot or monocot? If scutellum is present here, single cotyledon, right? What do you call this? Scutellum is present here. Is it monocot or dicot? It is going to be monocot. Scutellum is present in the case of monocot here, and if you notice here students, the entire radicle is covered by coleorhiza, RR rhiza. Here we have plumule. Plumule is covered by coleoptile, P and P. PP, coleoptile and plumule. Coleorhiza, radical, RR. RR. Okay, this one is a monocot here. Monocot here. Okay. You can see here, this is also an example here. Can you see? Endosperm is here. We have scutellum here, coleoptile, coleorhiza. Coleorhiza on radical, coleoptile will be present on the plumule, covering for plumule. Okay. Embryo of monocot possesses only one cotyledon. Obviously, it's a monocot, single cotyledon, and the single cotyledon is called as what? Scutellum. Scutellum. Okay. Coleorhiza, coleoptile, I told you already, coverings. Now students, seed. Now students, how many of you know that, right, a seed mainly has few main parts, right? Seed actually has an outer coating called as seed coat. We can see here, it has an outer coating called as seed coat. Now this seed coat can be of two types. Outer coat is called as testa, and we have tegmen. Testa and tegmen are the two parts of seed coat. Okay. Now in angiosperms, seed is the final product of your sexual reproduction, obviously, because entire ovule is converted to seed here. Okay. Now here you can see epicotyl, hypocotyle, radical, and cotyledon. If there are two cotyledons, it's a dicot. Single cotyledon, it's a monocot. Okay. Now students, a question to all of you, right? Now you will understand the concept of seed even better when I will show you a comparative analysis. Do you think seed is the egg of the plant? Like how we have in chicken. In chicken we get eggs. Do you think your seed is just like an egg? Tell me yes or no. Because if you take an egg, if it is fertilized, it can develop into a new chick. Similarly, do you think a seed is also like an egg? Now you will get a perspective, you will get a better perspective on what is the function of cotyledon, what is the function of embryo, what is the function of seed coat. Let me tell you, see this diagram will help you better understanding. Here on the left side I have a seed, on the right side I have an egg. Here, like how I have a seed coat, here I will have something called as eggshell. An egg will have a shell, here a seed will have a seed coat. Inside a seed, I will have something called as cotyledon. What is the main function? Main function of cotyledon is to provide nutrition. Similarly, in the case of an egg, I have albumin. Albumin and cotyledon are same to same. Now, inside the seed, I have something called as embryo. Yes, embryo is present inside the seed. Embryo is similar to this egg chicken embryo. So all of you are getting the comparative analysis here. Seed and the egg are almost same to same. Here seed coat, here eggshell. Here we have cotyledon for nutrition, we have albumin for nutrition. Okay, here we have embryo which develops into the new plant, and here we have a chick which develops into a new plant. So all of you are able to relate your zoology with botany and understand better. Tell me yes or no. All of you understanding better now? Is this makes more sense now to all of you? The function of cotyledon, function of embryo present inside a seed. So seed is just a shell. Seed is just an egg. Right? Seed is just an outer covering. Seed is this entire thing is called a seed, similar to an egg. Right? The main thing which is going to turn into a new plant, that is going to be the embryo present inside. Embryo present on the inside. Okay. Now students, remember nucellus? When I was teaching you about development of Megasporogenesis, I told you that when the embryo sac develops from the nucellus, and usually what happens as the embryo sac is developing, all the nucellus is gone. But in certain cases, nucellus can persist, and that is when we get perisperm. So what is the perisperm? Persistent nucellus is called as perisperm, and it can be found in the case of your pepper. Black pepper, and even in the case of beetroot, in the case of beet also, perisperm. See here. We learned this non-albinous, ex-albinous we learned here. Okay. The residual perisperm nucellus is the perisperm. Okay. Black pepper and beet. Black pepper and beet, we can get perisperm. Okay, perisperm. Now, there's a very high chance that what happens in the case of your plants is the seed is, once the seed formation happens, the seed is very dry. The seed is very, very dry because it loses all the moisture eventually, right? You see this. The micropyle remains as a small pore. Remember in the ovule, micropyle was a small opening, right? Where integuments were absent. That entire micropyle will remain as a small hole now. Remain a small hole, right? In a seed coat, this facilitates entry of oxygen and water into the seed during germination. For example, let's say here is a micropyle, the entire region there also we learnt. Hilum is the point of attachment of your entire, there it was funicle and entire ovule. Here we have hilum here and we have micropyle here. Now micropyle is a region where all the water and oxygen can enter inside. Okay. Seed matures, its water content is reduced. See, all the water content gets reduced as the seed is maturing. The general metabolic activity of the embryo sac slows down and it can cause dormancy. Dormancy is very important. Dormancy is a inactive stage in plant's life cycle. When the seed is waiting for the opportunity to revive again, as soon as there is water, there is moisture, there is soil, there is nutrients, the seed will germinate into a new plant. But how long this dormancy can exist? For example, students, there are plants called as Arctic lupine as well as your Phoenix dictifera. Both of these plants have the seeds can be dormant for 10,000 years. Here it is dormant for 2,000 years. The seeds are still viable. So they can stay dormant for very, very long time. Dormancy can be maintained. Very long time it can be there. Okay. Very long time it can be there. Okay. Now what is a fruit? Seed is done. Very simple seed. Embryonal axis. We learned about hypocotyl and everything. What is a fruit? What exactly is a fruit, students? Fruit is nothing but when the entire ovary gets converted to the fruit. And fruit mainly has two parts. A fruit mainly has two parts. It has something called as seed and it has something called as your pericarp. Pericarp is the covering. For example, pericarp can be of three types. When you eat a mango, students, mango has this outer skin kind of thing which you remove, right? Mango. That outer skin is called as epicarp. The mango flesh which you eat, nice tasty mango which you eat, right? The eating part is called as mesocarp. And the one on the inside which you throw away, that stony part inside, that is called as endocarp. So this is your entire fruit. Okay. Now students, there are multiple types of fruits actually. There is something called true fruit, false fruit. I'll tell you later on. Okay. Pericarp. Maybe fleshy, guava, orange, mango. Okay. It can be dry also. Groundnut is a fruit. Yes, groundnut is a fruit. Mustard, dry fruits. Okay. There is something called as true fruit and false fruit. What is the difference? The difference is very simple. True fruit will develop from ovary. True fruit will develop from ovary. False fruit, it will develop from ovary plus other parts of the flower. Okay. See here. However, in few species of such as apple, strawberry, cashew, thalamus also contributes. See, it's not only ovary has to be there. It is the thalamus also contributes to the fruit formation, or called as false fruit. Example is your strawberry, cashew and apple is a false fruit example. Okay, false fruit example. There is one more type of fruit, students. Parthenocarpic fruit. That is fruit without fertilization. There's one more type of fruit called as fruit without fertilization. That is called as parthenocarpic fruit. Without fertilization, there is a fruit. Okay, and these fruits are mainly seedless fruits. And these fruits are mainly produced because of hormonal changes. For example, enough of auxin can cause parthenocarpy. Okay, parthenocarpy. Significance of seed and fruits. What are the significances here? Dependable seeds formation, since pollination and fertilization are dependent on water. So seed formation means good. Okay. Nutrition to the embryo, obviously. Seed has cotyledons, endosperm provides nutrition. Dispersal of seed for various agents can happen. Seeds are important. Protection of the embryo, obviously. Sexual reproduction lead to greater variation. Yes. So significance of seed is there. Okay. So last topic is called as apomixis and polyembryony. Very simple concept. Very simple concept. What is apomixis? Formation of seed without fertilization. That is called as apomixis. Now students, the question arises here, how does the apomixis even happen? How is it even possible? How is it possible to get apomixis? The answer is very simple. No, NCERT doesn't tell you this. Unfortunately, NCERT doesn't teach you this. I will teach you today. There is something called as type 1 and type 2. Type 1 and type 2 apomixis. What exactly happens in the case of type 1? In the case of type 1, what happens? Understand, very carefully understand. Okay. What happens in the case of type 1 is that the entire egg is there, egg before fertilization. It undergoes, which is haploid, undergoes something called as endoreplication. Endoreplication, and this egg haploid will directly convert into embryo, which is diploid. That is called as endoreplication. Develop into the entire embryo. That is called as endoreplication, and this embryo will develop eventually and form the seed. So without fertilization we can get. Without fertilization we can get. Okay. That is called as type 1 endoreplication apomixis. What is type 2? In type 2 what is happening is, okay, in type 2 what is happening is nucellus, which is diploid already, nucellus will give rise to the seed. Nucellus, all the around the embryo sac is there, around the embryo sac, nucellus. The nucellus will get converted to seed eventually, or nucellus becomes embryo and then becomes seed. This same concept of nucellus becoming into seed is for polyembryony. What is polyembryony, students? Polyembryony, basically, poly means many. Many embryo can be there. Many embryo. Example is citrus. The oranges. Apomixis example is very simple. That is going to be your grasses and Asteraceae members. And Asteraceae members are the examples for your polyembryony. Okay. So this is the apomixis, two types, type 1, type 2. First one, the egg undergoes endoreplication to form embryo sac, that becomes seed. Type 2 is called as a nucellus becomes the seed eventually. That is the answer. Now here is a question to all of you. If you know the answer, tell me in the chat quickly, all of you. I have some questions for all of you now. Some questions on the board. I need to get my phone for seeing the chat properly. I can't see here, students. Very difficult to see. I want you to keep a track of your marks now, students. Every right answer 1 mark. Every right answer 1 mark. Okay. It's visible properly. Okay. Identify the correct set of statements. A flower of Vallisneria are colorful and produce nectar. Students, Vallisneria was wind. It's clear. I just saw in my phone. It's clear. Students, tell me quickly in the chat. Is A even correct? Valerianeria is water pollinated, right? Valerianeria is water pollinated, right? So this statement becomes wrong. It doesn't even need flashy things. Doesn't need flashy. Doesn't need to show off here. Flower of water lily are not pollinated by water, students. Yes. Obviously, water lily, it is not pollinated by water. Water lily is that, I told you no, mental. It gets pollinated by insect. Pollinated by insect. Okay. Correct. In most of the water pollinated species, pollen grains are provided by wetting. From wetting, there's mucilaginous sheet. Mucilaginous sheet. Okay. Pollen grains of some hydrophytes are long ribbon-like. I told you ribbon-like. Yes. In some hydrophytes, the pollen grains are carried passively inside water. Yes. Passively inside the water. It's carried. Okay. So this is the question. Answer is what? Answer is option number 3. Option number 3. Yes. Look at this question. Wind pollinated trapping, right? Wind pollinated trapping. Water pollinated, no. Cleistogamous, no. Impact inflorescence showing compact autogamy, no. Wind pollinated plant inflorescence showing with well exposed stamen. Well exposed stamen to trap, right? To trap the pollen grains. Trap the pollen grains. Tell me your marks now, students. If you tell this answer correct, it's plus 2 marks. Plus 2 marks if this is correct. Okay, plus 2 marks. Next question you see. The junction. Next question you see, students. The junction between ovule and funicle. Okay. This is the small junction. If this is a funicle, this is the ovule here. The small junction. What is junction called as? Junction is called as hilum. So, plus 3 marks. Plus 3 marks. Every question 1 mark. Every question 1 mark, students. Every question 1 mark. Not 4 marks, too difficult to calculate. Every question right answer 1 mark you get here. My class. Very simple. 3 marks if you're hilum. Which of the following represents the female gametophyte? Students, very easy. Quickly answer in the chat. Quickly answer in the chat. Female gametophyte. Female gametophyte. I told you. See, ovule is Megasporangium. Mega. Embryosac is female gametophyte. Embryosac is female gametophyte. Plus 4 marks if it is the right answer. Plus 4 marks if it is the right answer. Plus 4 if it is the right answer. The flower of Amorphophallus, I told you 6 feet height, 6 feet height, provides safe space for pollinators to lay egg. This is an example of. This is an example of amensalism, commensalism, anemophily, or it is floral reward. See, plant is pollinators coming in, pollination is doing pollination, but Amorphophallus is giving a space to lay the eggs. It is giving a reward. See, you are pollinating for me, I'll give you a reward. You can stay with me. You can stay with me. It is actually floral reward, not commensalism, not anemophily. So plus 5 marks. Plus 5 marks. Okay, look at this question. Look at this question, all of you. Look at this question, all of you. Okay. In angiosperms, haploid, diploid and triploid, structures of fertilized embryo sac subsequently are. You have to arrange them, which is haploid, which is diploid and which is triploid. See, synergid is haploid, yes. Antipodals are also haploid, all are correct. Antipodals are diploid, no. No, triploid. Synergids, no. Zygote is diploid, Primary endosperm nucleus is triploid, it is triploid. So what is the answer? Option number 4. Plus 6 marks. 6, no? 6 marks. 6 marks. Look at this question. Large, colorful, fragrant flowers with nectar acynian. Show off plants. Tell me the answer. Which are the plants which do show off? Plants which can do show off. Plants which can do show off. Quickly answer in the chat. Plants which can do show off. Quickly. Insect pollinated. That insect pollinated. So plus 7 marks. plus 7. Which of the following family set of families pollen grains are viable for months? Pollen grains are viable for months together. Months together. Okay. Leguminosae was there. Solanaceae was there. Rosaceae was there. This is the answer. So plus 8 marks. Plus 8 marks. Easy, students. Every question is so direct here. You don't have to even worry about anything. Very simple. Look at this question now. In angiosperms, the correct sequence of events leading to formation of female gametophyte in ovule is formation of female gametophyte. Okay. I am done. See, first what happens? Meiotic division in megaspore mother cell. MMMC is undergoing meiosis. We get 4. 3 will degenerate. So this is 1. This is 2. 3 will degenerate, 1 will survive. Okay. This will undergo 3 mitosis now. Mitosis 1, 2 cells. Mitosis 2, 4 cells. Mitosis 3, 8 cell. 8 cells. Okay, third option. Then migration of 3 nuclei towards each pole. Now there will be polarization. Polarization, they'll migrate. 3 here, 2 in the middle. Okay, so fourth one. Okay, formation of wall resulting in 7 cells embryo. Last point. So then what happens? Wall formation happens. This happens. So it is gonna be C B A D E. Option number 4. How many marks? Plus 9 marks. Plus 9 marks for me. Okay, next question, students. Identify the incorrect statement regarding pollination. Moths and butterflies are most dominant. No, students. It is always bees. Bees are dominant. Okay. How many marks was there, students? Plus 10 marks. Plus 10. Bees are more dominant. It is not this. Okay, next question. Cleistogamous flowers are invariably autogamous. Okay. Invariably autogamous. Okay. Cleistogamous flower is disadvantageous as there is no chance of cross pollination, obviously, because if there is only self pollination, that can lead to embryonic depression. It can happen. So that is a disadvantage. So both are correct. Plus 11. Okay. Next question. In the following set structure, correct A, B, and C labeling. A is seed code, obviously. A is seed code. This is gone. B is on the inside. C is the endosperm. And D is the hypocotyle. A is the answer. This is the NCI diagram, student. This one is a direct NCI diagram. I'll show you 1 second. See here. You can easily see here A is going to be seed coat, B is going to be cotyledon. B is going to be cotyledon on the inside. C is the endosperm. C is the endosperm. And D is going to be hypocotyle. D is going to be hypocotyle. Okay. Next question. That's all today. That is plus 11 plus 12. Plus 12. Let's call it as homework question. I'll give you more questions in the DPP format. Don't worry. Yeah, I just checked A's. It's not confusing. I just checked the. Okay. Student, this is a homework question. That is residual persistent part which forms perisperm in seed of beet or black pepper is what is it called as. Okay. So homework question in the comment section. And I think I'll stop the class today, students, because throat is giving up little by little. I don't know how come. Right. So thank you so much, students, for today's class. Have a great day ahead. Right. Hopefully you understood this chapter in a better way, if possible. Okay. So like the video, let me know in the comment section how was the class, because I believe we did in so detailed the Megasporogenesis, the entire pollination, outbreeding, artificial hybridization, seed, fruit, double fertilization, pollen pistil interaction, how it happens. So everything is done in detail today. Okay. So go through the video as soon as possible, because this month end the entire video is going to be out. Okay, I'm going to delete the video. Right. This is the only month the video is going to be there, unfortunately. Okay. So try solving more questions. And I've been telling you again and again, students, AITS. If you go to the description of today's video, students, I'm telling you honestly again and again. Okay. If you go to the description of today's video, right? If you can go here, right here, right? Here we have AITS offline, AITS online. Now, if you think you want more questions, you want to solve more questions, I'm telling you again and again. AITS is the must compulsory for every single NEET aspirant right now, every single aspirant. Because if the earlier you start solving mock tests, it's going to be better, right? So AITS is at 699 right now. You can see here, you can go to the description and you can see Shreyas here, say hi to him, okay? And you can buy it from here and it will be delivered to you, right? It will take approximately 3-4 days. They will reply to you, don't worry, okay? So get the AITS as soon as possible. And if you want to get a PYQ book also, there is a PYQ book available, I guess. Yes, here. This is a PYQ book. 37-year PYQ book. All 3 books is for 999. 9,000 rupees for 3 books. 3 books for 1,000 rupees, right? So this is going to be the entire chapter today, students. So get the PYQ books, get the AITS, get the notes, right? Download these notes, make your own notes from out of it, and keep studying, keep the grind on, and I will see you in the next class. Thank you so much, all of you. Thank you so much. Thank you so much, all of you. Okay, so bye-bye, take care all of you, and have a great day ahead.